Okay, well, welcome back. This is week 15 of E340, 542. Today, you're doing the lifting, you're doing the presentations. I want to remind you that by default, this is live streamed and recorded. If somebody would prefer that their presentation not be recorded, just say so and we'll stop the recording during that period. In general, I think people find that it's useful to have the recordings, but I certainly don't want to impose that on. So the order of presentation is Eden and Connor, and then Supriya, Dhanusha and Poorva, and then Peter, Zachary, and Gabriel, and Joel, and then Lindsay and Grant at the end. Supriya, Peter, and Lindsay have said that they want to do a second round of presentations. Please do make sure that during your presentation you share a link to your NanoHub app if it's published, or a downloadable version of the notebook if you haven't done it yet. Remember that to get a final grade for the class, you have to have published your NanoHub app, so make sure that that's done. And then also, please do fill out those comment sheets and get them back to me. I'll cut and paste them and send them around to the teams for help in their final presentations as well. Any questions or comments before we get started? Okay, we'll start out with Eden. You want to take it away, please?
Yeah, gladly. So Connor was actually good enough to record a sort of introduction to our project from Singapore. So I'm going to start by playing that for y'all. And then I'm going to walk through the notebook and answer questions. So I'm going to share my screen and let Connor do some talking. And if for some reason you can't hear or see him or whatever, let me know.
Hello, everyone. Due to the 13-hour time difference, I am either in the future or the past, depending on whether you want to think about time zones or when I'm recording this. I'm sorry I'm not able to be there with you all today, but this is kind of my attempt at being as present as I can for the push at the end of the semester, and I look forward to watching the recording of everyone's applications and being able to offer some feedback. But for now, I'm here to talk to you all about mining Eden's notebook application for numerical methods for dynamical systems theory in ordinary differential equation models. Generally, whenever I am approaching a novel topic, I find it really helpful to have a broad global overview before diving into the more gritty details of the code or the math. So that's what I'm going to try to do in my section of the presentation here, to give you an idea of what we've been trying to accomplish and where we're going. And then I'm going to let Eden dive into the specifics of the notebook and other details, in part also because due to how this format is happening, I think it'll be easier for him to answer those questions than me. But okay, let's dive into the presentation. Okay, so broad outline of this is going to be trying to pitch why we want to use dynamical systems theory for tackling the types of models that we've been looking at in this class. And very broadly, I'm going to offer an overview of phase, fortress, and bifurcations. I'm then going to switch gears and talk about why we want to have a numerical methods toolbox, because when we've seen plots like these in the past, it's been largely by product of working with the equations by hand analytically. And like I said, then I'm going to pass it off to you. So general goals. This material was really meant for people who do not have an explicit background in dynamical systems theory.
Sorry, I think he's muted for me at least.
All the
In
things
here.
that we did, but I'll kind of
Sorry, I had muted myself. Did that mute the video too?
Yes.
Okay, let me go back a few seconds. Sorry about that.
good introduction to kind of some general concepts and specifically surrounding equilibrium behavior. I'm not going to be able, I think a relatively thorough notebook. I'm not going to have time to get through all the things that we put in, but I'll kind of try to cover some of the core ones. I think another hope we have is that it will help folks understand the richness of OBE models, both in terms of the analyses that can be done on them, and also in terms of the complexity of behavior that can be produced qualitatively by them. We also wanted to demonstrate this all as a Python implementation. So Ian and I both generally work in Mathematica, which is an amazing and powerful piece of software. What we've done here does not cover at this time what we are able to do over there. But the difference is that Mathematica is a corporate piece of software, so it's pay to play, and we wanted there to be a good resource for people who wanted to get involved with this type of work that only rely on kind of the core mainstream Python dependencies. So, okay. Why dynamical systems theory? So generally, when we're looking at dynamical systems, we're, to borrow Professor Glazier's saying, trying to deal with dynamics on networks. So we're explaining the time-changing behavior of a system. And this can be broadly separated into two classes: the transients of the system and the limit sets. So if we're looking at a transient behavior, it basically implies that starting from an initial condition, we're going to see some type of structure to the trajectory, but it will die out. A member of the limit set, on the other hand, implies a region in the states of the dynamical system where if you start exactly on it, you will stay there. Now, if you step or are perturbed slightly off a member of the limit set, what's going to happen? It's going to change according to stability, which we're going to talk about. But let's not get too far ahead of ourselves. And broadly, by piecing together this underlying structure to the dynamics of the system, you can begin to tell a story about the behaviors that are available to a specific system or a class of systems as you change their parameters. So really, quick reminder, maybe not a necessary one, but the most common way to look at changing these types of systems is through a time series. So on the x-axis, we have time, and on the y-axis, we have the value of the state variables. We then have a line, a curve for each of the states that we're looking at. And this provides a very nice way to capture both time-dependent details. So for example, we can look at the slopes. And it also provides a two-dimensional way to look at many state variables at once. What I'm about to show you is a more graphical method for lower dimensional systems. And I'm really focusing on that because that's Oh, sorry, that was a timer. Just keep me on track. And of course, the timer got me off track. Right. So basically, we are offering another powerful way to look at these things. Specifically, I'm going to be focusing on 2 dimensions because we dealt with a lot of those models in class, especially pre-going into feed-forward networks. So, general reminder, this is a flow. So basically what we've done here is that instead of looking at time, we've plotted for a 2-equation system, 1 state variable along each axis. And we're trying to understand what types of changes we see. So if you recall in some of the work we've done in this class, we've had moments where we say, okay, as we start from different initial conditions, what happens? This provides a very holistic way of looking at that. We can actually look at all these different initial conditions by looking at different regions in the space and follow the flow to see what's happening. And we can start to interrogate it for details that we might want to investigate. For example, in this one, I'm hoping you can see my mouse, but we see, of course, that a lot of arrows are pointing towards this kind of center point here. But then something's happening when we get really, really low in the values of the states, where things seem like they start to turn inward. So this tells us that somehow it seems like there's 2 regions that the states are tending towards, and there has to be something separating them. So as Professor Glazier introduced, one way to approach this is null find. So if we have 2 equations in the system and we set them equal to 0, we can then look for the regions in state space where one equation is has no rate of change and carve it out. So like, let's take this orange line here, right? As we can see, pretty much all of the trajectories that are around this orange line only have behavior relative to the y-axis variable, right? If we look here, they're pointing straight up. If we look here, they're pointing straight down. There's nothing going left to right along the x-axis. So that's one of our null finds. And then we see the same thing here, right? We see behavior that is essentially going to the left. We see behavior right here that's essentially going to the right, and nothing really happening in terms of the y-axis. So these are cases where the rate of change for one of the state variables is 0, but the overall time evolution of the system is still ongoing. In order to look for cases where we are at equilibrium, we have to look for where both are 0. So that's where the null lines intersect. And as we can see, we have 1, 2, 3. And if you could recall from before, we saw just from the flows that there were kind of specific regions that the system was tending towards, and there was something dividing them. And this tells us that, okay, the thing that divides them is probably going to be right here. And then these are probably the exact points of where the system is moving towards over time, depending on its initial condition. We can then test that and actually solve for the equilibrium by putting the value in. So there's a few ways we can do it. One is that we solve it analytically, like we did in class, or we can solve it numerically. And we'll talk about why you would want to do it one way or the other. But basically, we can see here that the equilibrium values do correspond exactly to the intersections of the null lines, which is great, but our story isn't done, right? Because we have this moment of saying, okay, well, we have 3 equilibria. Why on earth are they headed towards 2 of them and ignoring the other? So that involves doing what's called stability analysis, which we give an introduction to in the notebook. it's a little bit too extensive for me to do given the time constraints on the slide. But basically, we say if we perturb off of the equilibrium point, what's going to happen? And what we see is that we can classify these points differently. So the two that the state variables are moving towards are attractors. Basically, if you push slightly off of it, you're going to just head right back towards it. And we classify those similar to how our advisor, Randy Beer, does and color them blue. The one that separates them is called a saddle node. And that is quite unstable. You'll have trajectories that head towards it or away from it due to things that we're not going to have time to talk about. But generally, you're never going to fall on it. As the trajectories approach it, it's going to push one way or the other. And what this equilibrium point does is that it carves out the basins of attraction of these two blue points. So it's going to carve out the space of initial conditions that will head towards one or the other. So this gives us a lot of information about describing the actual possible behaviors of a specific dynamical system. But there's also families and classes of dynamical systems. And one way to start to get at that is through bifurcation. So here we have an example of a 1D system with state variable X and a parameter that's being varied. Same as before, the stable equilibrium points are colored in blue. Here, though, we don't have a saddle, which was colored green. We have an unstable equilibrium point. And what that implies is that no matter in which direction you perturb it, it's going to move away. There's not going to be cases where it heads towards and comes close to it and then keeps going off in the other direction. Things are just going to be repelled from it. And every slice along this parameter is a different type space portrait. So if you were to take a vertical slice through this space, you would have a state space. So what this tells us is that for different values of parameter C, we only have one stable equilibrium point. And then there's this very special type of point that is technically called non-hyperbolic, but you can look at the notebook for terminology. And what this shows is that there is a case where the system is changing in a very meaningful qualitative way. So now instead of having one equilibrium point, we have two where their basins of attraction are separated by an unstable equilibrium point. So there's a story to tell in all of these cases, and especially for these low dimensional systems that can be told in a graphical way, which is wonderful. Okay, so more in the notebook, but that's kind of the broad overview of the tools that we're trying to offer. In terms of numerical methods, generally they're pretty computationally expensive, and also having to work your way through state space can get difficult, especially as the dimensionality of the model increases. But it's also not typically possible to solve very nonlinear systems by hand. So you actually need a numerical methods toolbox to generate some of these pictures and to investigate them. So in class, we got a good introduction to how to draw a picture of phase space using the analytical methods. And here, like the pictures I showed you in the last section were actually all cases of numerically generated phase spaces from a system that I've been playing with for a while in Mathematica, and it's the degree of nonlinearity is quite unfriendly. if you're going to attempt to do things by hand. But so if you're going, and the truth is nonlinearity is the norm in the natural world. So if you are going to be trying to model biological systems and you want to do these types of analysis, it's really, really important to have a toolbox where you can both approach it analytically in case there is a clean enough solution, but also pivot if need be. Yeah, so once again, I just wanted to show here and stress that although this might look pretty similar to some of the plots that we generated in class at one point, and although, sorry, jet lag, it might look similar to what we generated in class, and the philosophy and underlying methods might be similar. This was made using numerical approximations for a system where approaching the production in the way that we did would not really be tractable. So our notebook, once again, really attempts to do two things. It tries to offer the start of such a toolkit while assuming minimal background knowledge and offering resources to fill in the gaps. And it also tries to do it in a way that is not dependent on some of these software packages that are either very expensive or not very well supported. Yeah, so with that, I'm gonna sign off. Hopefully that was coherent. Hopefully I can find some caffeine soon to counter the jet lag, so I apologize if it wasn't. But yeah, all yours, Eden. See you, everyone. Good luck.
Okay. So with that, I'm going to take over and pretty much just walk through the app in brief. I'm happy to answer any questions about how we pulled off anything in particular if people have them. There was a bunch of pretty fun puzzles to figure out how to make Python do these things. But overall, here's our splash page. Overall, we were interested in making this an educational tool as well. We got some really good advice from Hayden too, from meeting with him a week or two ago. So right now we've sort of laid out some of the learning objectives that someone should be able to walk through this notebook and basically walk away with. And Connor kind of described a bunch of those in that video, especially because he sort of tried to teach some of those learning objectives along the way too. But basically, we want to familiarize people with dynamical systems, understand the application to ODE modeling, and then sort of be able to use these tools we've built in Python. And so this is what the notebook looks like right now. So we've sort of laid out this introduction again of what the learning objectives are. We've got some disclaimers about you might need to know some things about calculus and about like using linear analyses and stuff like that so you don't just get sucked in halfway through and then be like, oh my God, I don't know what any of this means, and then you've wasted time. So we start with some of that and some of this sort of background on what ordinary differential equations are. I'm going to sort of glaze over this for the sake of time. Along the way, we've implemented a lot of these intuition check questions, which are basically drop-down widgets that are meant to show, that are meant to just test you along the way. There's no formal assessment of how well you're doing or anything like that. I haven't loaded my packages, so I can't show one. But this is sort of our interactive version throughout, because it is a bit of a long notebook to sort of just read everything. And we felt like these were pretty important to keep in there, right? So we've got a lot of these questions, answers. Right now, I'm showing this in the editing mode. The one outstanding thing we need to fix is how to show some of the code cells in the app and not show others. So I'm going to wait and ask about that at the end, because that's sort of our last little thing. But for now, I'm showing it to you in the editing mode, because obviously you can see all the answers to these questions if we leave it like this. And then we get into some of what Connor was talking about. So these are using tellurium models. We wanted to try to mesh both together as best we could. So some of these examples are shown in tellurium. Our first state space example he was talking about is also shown using tellurium. And this is A-B space, right? So this isn't a time series. This is a trajectory. We give some rules about what trajectories tend to look like. So simple rules, including trajectories never cross each other. There's never one point in space that points in two directions in these models. It's what makes them entirely deterministic. We talked through some of that. And then there's two big sections, one of which is one-dimensional systems, one of which is two-dimensional systems. And basically, in both cases, we lay out a system of equations. So in one dimension, it's one equation, in two dimensions, it's two. And we walk through visualizations of the time series and then some of those graphical methods kind of mentioned. So here we're looking at a system that was shown, that we'll see the bifurcation again. This is the same one. And it's sort of showing this time series of all these initial conditions of this one state variable, and see, oh, they end up in two places kind of. And let's talk about that and figure out, how can you figure out where those places are, kind of things like that. And we talk about the analytical solutions. So we talk about doing the algebra to figure this out by hand if you want to. And sort of introduce early ways of finding it numerically. This is a very basic cut dry one. And then we're like, yay, you found your equilibrium points. Then we get into the stability analysis stuff. The stability analysis stuff has a bunch about the Taylor expansion, which is still complicated and makes my head hurt, and all these other things about linearizing around a certain point to basically answer what Connor was saying about, if I take a slight step off of this point, where do I go? And we have some intuition checks about this as well. We show how with unstable points, which you're not going to get in this kind of time series, how if you run time backwards, you can get the other point. And then we are also showing, then we get into the family of equations, as he was saying. So if I add this parameter, we can see the behavior of the system change. And then here's the code for this bifurcation plot. So this is our first significant, like, here's a thing you can play with. Here's a tool to use. It's using SymPy a bunch. So this one is, and Connor said all of them are numerical. This one is a little bit of both in terms of actually plotting this bifurcation, because SymPy is basically an algebra engine in Python. So then we introduce this bifurcation plot, as Connor mentioned. Have some more intuition questions, because bifurcations are not very intuitive, and making all these mental leaps between these graphs is hard. And then we do two dimensions. Two dimensions, very similar. We talk about state space. We talk about trajectories in state space. And we talk about nullclines here. Here's another big tool that generates this flow and nullclines superimposed on one another. We have some more questions. And then we start to introduce a lot of the numerical root finding, which is the numerical version of doing the algebra to find where the equilibrium points are. And this comes in a few steps. So this one is one that Connor built, which is basically you give the system some guesses. So you look at a picture like this, right? Because this is generated for you. And you look at a picture like this and you say they cross at about -1, -1, about 0, -5, and about 1/2, 1/2 kind of thing. And you would give the computer those guesses. And what it does is it basically, it iterates from there and sees where the points end up, and tries to identify fixed points in that way. And that's like the numerical method he's talking about. And so this spits those back out. Then we talk about stability in two dimensions, which is a bunch of linear algebra, including the Jacobian matrix. We outsourced a lot in this notebook as well to other video lessons, because going through things like that is just far beyond what we could fit in here. And then we introduced some functions for how to do that. So turns out SymPy has some great stuff for linear algebra too, in case anyone is interested in that. So we can get this Jacobian matrix out of it, talk about all this matrix math you would need to do to basically get these eigenvalues, which tell you something about stability. We have some questions of trying to solve for those things on your own, as we are still analytically talking about how to do it as we go through this. And then we have some code that will tell you the stability for you if you are like us. And once you know how to do it, it's really laborsome to do it for every single thing you look at over and over and over again. So this will do it for you. And using that information, we can amend this picture to add the stability information, like Connor was saying, to the fixed points that are found here. It's color coded again according to this blue, green, red scheme that Connor mentioned. Then we provide sort of this unguided example. This is just a two-dimensional system, and we've just asked a series of questions for people to figure out analytically or using code. So we've given a little bit of starter code, like some hints of how you might want to approach these questions, which basically gets from equilibrium points all the way through the stability. And we have this separate notebook that's linked to the same GitHub that has the answers. We were recommended to not put the answers in this notebook, as it's late enough in the notebook that people would probably just look at them. So that's where those are. And then we provide some other resources. So some textbooks, including Herbert's textbook, and then some other websites and YouTube channels that we are fans of that are sort of related. So that's what the notebook looks like. Connor and I are hoping to eventually turn a lot of this into a Python class at some point with a bunch of added stuff. I mean, like this is a early cut into the kinds of things that we are able to do in Mathematica, as he said. And I think we would need to flesh it out some more. But yeah, it's meant to be an educational tool. It's meant to be a precursor to some other things we're hoping to build too. And for anyone who needs to do this stuff in Python, there's some starter code in here. So that's what we got.
Okay, that's great. Does it run now?
Everything runs. Yes, everything runs in NanoHub. I checked it all this morning. The only bug per se is the showing the code cells, but everything should run in here. All these widgets run now. The simulations go. The thing that takes the longest is the bifurcation plot. It takes about 30 seconds, which all things considered is not that bad for this kind of thing. So I can show you, like, this should spit it out. And we've sort of tried to comment the code too, so you know where you need to input things and what you need to do. Trying to balance between like providing the code as a tool and not having it be parsable at the same time is tricky. So this is the one that takes the longest, but everything runs fine. This one was freaking me out earlier because I forgot to have a plot show. It was very exciting. That was my biggest bug that I had to deal with the last 2 days.
I'm trying to remember. I know there is a tag for showing or not showing cells. Juliano, we have to help them find that.
Yeah, I was doing some searching this morning, but
that's impressive as an educational app. I mean, you've done a lot. I mean, finding unstable fixed points isn't easy. Finding the null lines isn't easy.
Yeah.
And so at the moment, if somebody changed the equation, what would happen?
yeah, I can give you an example of that once this actually plays out. I think the best visual of that is in the null lines, right? So if I were to have these ODEs here, this is where you sort of define your equations, and then this takes over for you. As long as you've defined them properly. Let's see if the bifurcation is running. It is still running. It's being slow. It's okay. So this is the example given in the ODEs above, but let's say, I don't know, I wanted this function to just equal x, and let's make this one be, what's something that would be interesting? We could
So
just,
why don't you just leave the one you had before? Leave
yeah,
the
sure.
And then just add, I don't know, acquired it to it, y to the y, x times y or something to one of them.
Sure. Here, I need to, this is strange. I promise this bifurcation stuff does work because that's how I got it to load in the first place. But for some reason it's,
Well, when you're being watched, these things always,
no, Zoom makes everything 10 times as slow. This is what I've learned.
yeah, I've noticed that.
Restart it. I'll pop up here. But this is also, this is up on NanoHub too, so we can send it around if people want to play with it. And if people run into anything that doesn't work, like you're saying, that'd be good for us to know too. Okay. So this is the original plot once it loads, which is now conditional statement.
I mean, I noticed when I was doing null lines for class that
Mhm.
they're really finicky.
Yeah. So let's say, so here's this graph, right? It's the one you were just asking about. Here's the original one. Let's say we add, like you're saying, just add y times x to this. And then we'll plot this again. And it's going to take a second. It would be nice to keep it side by side. It does kind of overwrite what's already there, but that's something to keep in mind for the future. And now it's, so this purple line has moved up a bit. It's, I think actually what happened is it got compressed in a way. The y times x kind of bent that bottom up a little bit.
Yeah.
Really where this would be better reflected is probably if I was to come down here and show you the change in the phase portrait, which I think I would need to run a bunch of code beforehand to do. I can try. No, it's mad about something. No. It does need to be run in order. That is something important. But it should be adaptable to the idea is that at the end of it, you should be able to take these code chunks, take them out and put in a system of your own interest. You should also be able to put the example system in there too and have that work for you as well. So why have you not defined this? I just asked you to. Yep. Let's see if this one's changed too.
Other comments or questions? I don't mean to monopolize the. Peter,
Yeah.
go ahead.
Yeah. So you mentioned earlier that one of your goals was not only to create an educational app, but also to try to implement it using some of the core Python packages. I think there's 4 of them. Can you talk more about that and your implementation? Did you use
Yeah. Yeah, absolutely. Well, that's running. I can gladly do that. So the big things we're drawing from in Python, Matplotlib is just for making pretty pictures. I think we actually dropped Optimize. Optimize was one way we were trying to approach the bifurcations earlier that I think has managed to stay in there. SymPy is the only other significant use case here, and that's in the bifurcation plot. So here I can try to dive into some of the specifics. Right, so this bifurcation plot here, what it's asking for, originally what it asked for was for you to compute the derivative yourself and then plug that in. And we've since changed it to to take the derivative for you, which is a lot of what this SymPy stuff is doing. Basically, the SymPy is looking at, this is a computation of the derivative where you give it the symbols of these equations that you define, where are the equations defined here. So this is using like lambda notation in Python, which is a pretty standard way of defining equations. And you plug those in here, and it has this differentiating function that it can give you the derivative for you, and then you can turn that into an equation back into this lambda phi is basically give it back as a lambda function in Python. And then sort of you're trying to find these branches here. The branches are, if that had ever loaded, it's right. It's this S curve. It's the blue, red, blue. This is plotting them. This is solving for them. So it's asking, like, it's taking a bunch of vertices and asking if that derivative at the vertex is greater than 0 or less than 0. That's a way that we can determine stability. Has to do with the sign of the derivative. That's just part of the analytical method as well. And then it's plotting it. That's most of that one. The flow is actually really nice. There's just a matplot function that's called streamplot that'll do that for you. It's really great. And there is some more going on in here in terms of the equations, and you'll see a lot of this is Connor playing with the tolerance, which has to do with how close to 0 do we have to get to count it as being 0. In terms of if I was to move away. So in terms of computing the null clients, that's really important, which comes in down here. It's basically saying, because it's sampling the space a bunch and then asking at which of these points am I basically not moving. And then, so if I set that tolerance really low, you'd get just these big blobs of color in there. And if I set them to, if I make them too specific, I won't get anything. So this is kind of one of the uses of the tools that gets tricky. It's trickiest with the bifurcation plotting, actually, in terms of sort of these tolerance levels and things like that. But that's the graphical stuff, and then the root finding and the stability happens a lot in here. This is the root finding. Again, has this kind of tolerance level setting. And you have to provide it with guesses, right? Like that's the important thing is you have to provide it with guesses, and then it's going to iterate until it decides it's not moving anymore and it's going to stop. The stability is using information from the Jacobian. I could get into an analytical discussion of how that works. It basically has to do with the eigenvalues and if the eigenvalues are positive or negative and in what combinations. So this is just a conditional statement talking about, hey, let me look at the eigenvalues and see what they look like. And the eigenvalue stuff is using SymPy, I think. Or it's built in. No, eigenvalues this is actually built into Python, this function. And then all this is sort of plotting that on top of it. It's just all it is hey, I'm going to make this point out of the roots I found and color them based off of what this function told me here. So that's my quick and dirty of how that all works.
It was interesting. So thank you for going into more detail about that.
Yeah. And it appears our phase portrait is finally generated down here. So, ta-da.
Any other comments or questions? I think it's a great notebook. I think if people will use it. It wasn't quite what I was expecting, but since I didn't have time to cover a lot of this material in class in detail, it's nice to have you guys come back and do that. Appreciate it.
Well, yeah, we did sort of formulate it too around what you'd exposed us to in class and being like, what would be a way to present this in like an hour total? So,
well, when I was doing those plots, finding the null lines was a pain. Herbert did it in Herbert's textbook. He did all of the null lines analytically. and then just plotted them. I tried doing a few numerical ones, but boy, you need very tight tolerances because finding zeros is so that's
It's an interactive thing, and it's tricky because like you can't just blindly implement these things. I think some of what we found with the Mathematica packages we use a lot is you can actually, it's dangerous because you can sort of get comfortable and then it'll make mistakes and you won't notice. This is much more hands-on where it's at right now. So,
well, as I say, I remember in the old days, Mathematica, of course, it evolved a lot in 20 years. But 20 years ago, in Mathematica, you give it partial differential equations and say solve, and it would give you something.
mhm.
And it wasn't always right. You had to learn to have pretty good ability to determine whether the results were sensible or not before you got to carry it away.
Right.
Okay. Other time for one last question, or anybody else? Okay, well, thank you very much. I can't say thank you to Connor directly, but I appreciate his doing the
pass it
report.
on. Yeah.
Okay, great. Okay. So I think next on our list is Supriya. Are you ready to present?
Yes. Yeah. Let me share my screen. Okay. Hi, we are team of three, Dhanusha, Poorva, and me. So our topic is computational model of EGFR and IGFR pathway in lung cancer. I would like Dhanusha to go forward from here.
Yeah, thank you. So this is an educational application that will help us to understand the activity of EGFR and IGF-1R in the activation of ERK pathway in terms of cancer. And you will hear like a lot of new variables and a lot of new gene names, but it's quite easy to understand if you understand the basic pathway of how it works. If not, then you'll feel like it's a lot of new things and a lot of new concepts, and it might be a little bit confusing. So just make sure you follow how the pathway works and how the variables are related to each other. We'll try to keep the pathway alongside throughout a presentation so that you can go back to it and refer whenever you have like some doubts. And what we have done here is we have tried to replicate a paper that has already been published, and we thought we will add like a couple of more plots so that the understanding is better. Next.
Sorry about that.
Yeah. Okay. So the first one is EGFR. EGFR is epidermal growth factor receptor, and it belongs to the receptor tyrosine kinase superfamily. And this is a membrane receptor that is like in the surface of the cell, and it plays main role in embryogenesis. That is the process of formation of an embryo. So the main role of EGFR is for cell proliferation, cell division, and then the cell growth and its survival. In case of adult tissues, it helps in the same. And in case of when there's cancer, there's a couple of mutations that's taking place in the body, and then the EGFR is expressed more, and they help in the growth of cancer cells as well. So in case of lung cancer, which is predominantly the most that's the cancer type which is abundant, like which is the first type or something. So EGFR is expressed like 40 to 80% more in case of lung cancer. And And it is a ligand activated receptor. And whenever there is a epidermal growth factor or like transforming growth factor or like some other ligand that is compatible with EGFR is bound to it, it gets activated. Once EGFR gets activated, it leads to the activation of so many other metabolic pathways and the trans-signaling pathways of the body, which are really important for cell survival and the cell's growth. So it activates many pathways like the ERK pathway, the PI3K pathway, SRC, JNK, JAK-STAT, and there's like a couple more. So in case of JAK-STAT, it helps in like the inflammatory response of a cell and the stem cell maintenance and all these things. So every pathway has its own significance and has its own role and how important they are. But in our project, we are mainly focusing on ERK pathway because the paper that has been published is working on ERK pathway. So we thought if we stuck to ERK pathway for the first, we will have we'll have something to refer to and see if we are going on the right track since it's a new topic for us. So we are going to be focusing on the ERK pathway. And the next main factor, the next main gene that we are going to be focusing on is the IGF-1R. It is insulin-like growth factor receptor. And when EGFR helps in the division and the proliferation, the IGFR helps in the transforming events, that is the cell differentiation and then making it into a specialized type of the cell, like narrowing its function. And similar to EGFR, this is also highly expressed in case of the cancer tissues and then the cancerous regions. And it has a special function. It is anti-apoptotic agent. That is, apoptosis is a function that is undergone by a cell. Apoptosis is nothing but cell death. So whenever a cell matures and when it's about to run out, it undergoes the apoptosis function and then it kills itself. But in case of cancer cells, the cells don't die. So the population of the cells keep increasing and then it keeps stagnating, and that's how cancer tumor and everything is being formed. So this IGFR, it does not promote apoptosis, it prevents it. So it really helps the cancer cells to grow faster and then invade the body. And yeah, so in the early stages of cancer, you can see a significant increase in the amount of expression of EGFR and the IGF-1R. And now now that I have told about the two genes, I'll tell you about why, in brief, what the ERK pathway is. So ERK is nothing but extracellular signal regulated kinase 1, and it activates so many transcription factors to regulate gene transcription. So transcription is the process of where the DNA is getting converted to mRNA, that is the messenger RNA, and the mRNA will lead to the formation of proteins. So if the transcription goes wrong, then your protein formation is going to go wrong and is going to affect a lot of a lot of functions and the daily activities and so many other things in different pathways. So the core of this pathway, so to get the ERK to be activated, the RAF, MAP, and the RAF, MEK, and then ERK has to be activated, which is activated by the EGFR and the IGF-1R. And so this is the pathway that I was talking about. So this is the overall pathway of which we are going to be working on. EGFR and then IGF-1R is the main are the main factors that we will be talking about. And then it activates the SOS, and then it activates RAS, and then the MAP kinase. We'll see all these things in detail, and you just need to keep this pathway in mind. And yeah, can we go to the notebook, please? Supriya?
Yeah, sure. Yeah. Let me unshare my screen and then share it again.
Sure. Problem. So I guess whatever I said right now would have given you like an overall idea of how the pathway works, just to give you like the importance of it. And now we will see like in detail how each factor affects it and how they're all like dependent on each other and its function.
There you go.
Thank you. So this is our notebook. We have given a little bit little bit background of what the gene is and then how the pathway works and everything. So these are the mathematical ODEs that we will be using, the kinetic reactions that are used in the models that we will be implementing. And we found this, this was from the paper, this was the ODEs were already published. So we took the ODEs from the biomodels and then we just imported it in our notebook, and this is what it is. Can you go to the wild? Okay, yeah. So the first plot that we are showing here is the wild type ERK. So wild type ERK is nothing but how the time response of wild type ERK. So this is this graph shows how the ERK responds in the normal concentration of EGFR and IGF-1R. We did not change anything in the initial concentrations. The initial concentrations are given which is equal to the concentration that is found in a normal cell in a normal environment. So this is how the wild type ERK response curve is. And as you go down, our next plot, it shows, okay. So it shows how the EGFR and IGF-1R receptor behave as the time goes. So initially, the EGFR concentration is abundant in our body and the IGFR concentration is low. So when you increase the concentration of EGFR, we are trying to find how they both react to each other. So if I keep increasing it, they still, so even though the concentration are like the same, they don't get expressed at the same time, and then they both are not activated around the same time. That is because the EGFR concentration is abundant in the body and then IGFR is low. So when we gradually increase the EGFR and IGFR, we are trying to find how they react to each other. And at one point, the concentration of both of them, like at one point, the concentration, the expression of both of them become equal at a certain point. And we are trying to see if they inhibit each other or like promote each other, like when the concentration changes. I don't know if it makes sense. We are seeing how they both affect each other by changing its concentration. For example, if IGFR is going to reduce or inhibit the EGFR's reaction, or the vice versa. That is what we are trying to see in this plot. And then we go ahead and then plot. Can you just keep the plot, please? It's just
use the cursor. Like
yeah, my cursor is moving too much, so
I'll work on that.
yeah, thank
Just
you.
let me
Please.
know where to.
Yeah, the wild type one, like the next plot 3.
Okay, 1 second. Oh, okay. So here we go.
Next, yeah, go to the plots.
Mhm.
Yeah. So in this plot, what we see, so we tried to plot the activated ERK's time response for different concentration of the EGFR and IGFR. The first one is the wild type, that is a normal concentration in the body without changing anything. And then the next plot is the active Yeah, so this is like EGFR and IGFR down expression. When both have, when the concentration of both EGFR and IGFR are like comparatively lower than the normal condition. And this one is for the overexpression. And then we see when one of them is overexpressed, that is when EGFR is overexpressed. And then the next case is going to be both of them being co-expressed equally, like both of them being expressed too much. So overall, if you see the graph is going to look the same because its activity is the same. It's just that how much time it takes to react and how much time it takes for the ERK to get activated. That is the only thing that's going to change. So that is going to be changing. And then you can notice the concentration is being changing in the y-axis when I change the graphs. So that is one more thing. That is because you're changing the concentration. So you'll see like the activators get like, based on that, the amount of activators, amount of EGFR, amount of ERK that gets activated changes. And then finally, we have this graph, which shows all the type, and you can see the comparison of it. So if you see here, if we see here, the co-expression has the least time. Yeah. So when it is co-expressed, it takes like less amount of time to for the ERK to get activated. That's what it means. So you can see like when each of them are getting activated here. And now that we have seen like the overall idea of how they all work, now we'll try to go into the main, like every individual component of how the SOS, the MAPK, and the RAS and other genes affect the ERK's activation. And Poorva will take over from here.
Thank you, Dhanusha. Supriya, can you just take me to the model?
Okay. Sorry about going back and forth within the code.
Like it's not, the picture is not like clear. How do you decrease the size?
Okay, 1 second. Let me just decrease. Yeah.
Yeah. So we, like Dhanusha explained, the whole process of EGFR and IGFR working together. So while understanding how they lead to the activation or formation of ERK, there are a lot of little units which are involved in it. So the first one is SOS. SOS is the most important component as it is involved in the activation of MAPK pathway, which in turn activates the ERK pathway. So, well, let's go to the
Code. Yeah.
working.
Yep.
So, right, can go to the graph. Yeah. So here we know that after the ERK pathway and IGFR, they make SOS. And when SOS is present, it is basically active. When there is the dissociate SOS present in the nature, it forms and it activates the ERK pathway, and that can be seen by like if we see the initial value of AS. Can you go to the 0?
Yeah.
Yeah. So ERK is normally working when ASOS and it comes in the environment of DSOS. DSOS is basically the dissociate SOS, and it dissociates the initial ASOS value. It starts to increase. So when it starts to increase, the ERK activation happens. So after, when the ERK, the SOS is there in the environment, it further stimulates the formation of RAS. And what RAS does is RAS further activates the ERK pathway, the MAPK pathway. So, well, moving forward to the next one.
Yeah.
Yeah. So when ASOS, it activates the RAS. What it does is, so this graph basically explains, can you side by side give me the path,
Okay, let me try that out.
I'm sorry, because I don't think so it will be clear if we don't have
I
the
have
side
to
by side.
reshare again. We are really sorry about this. Okay. And yeah, is it visible, both the screens?
Yes, it is.
Okay.
So see, when we, okay, I can also. So when SOS, it stimulates the basically exchange of GTP and GDP. It activates the GTPase RAS. So what that does is it activates the MEPK pathway, which is visible here. And along with that, we can see that EGFR and IGFR, they also create PIK3. So PIK3, along with when the RAS is active and RAS is activated, so what it does is it creates an AKT. AKT is basically a negative inhibitor. What it does is when ERK pathway is activated, some concentration, when concentrations are increasing, ERK pathway will increase, the cell proliferation will occur. So AKT, what it does is it inhibits. As the concentration increases, it will keep on inhibiting the pathway, and a time will come when RAF will totally like vanish out, and the ERK pathway will stop. And further, that can be explained here when, to the next graph, AKT and RAF's activity. So AKT and RAF's activity, when we look at that, just go to the initial value, 0. So yeah, as it is, yes. When AKT comes in the environment, there is already RAF is there. So as the concentration of AKT increases with time, RAF's activity slowly decreases. So when it keeps on increasing, a time will come, RAF's activity will totally decrease, and AKT will start acting itself. So going next step to PP2A. So PP2A was like not mentioned in anywhere in the paper of its origin, but it is present there. It's present through like another pathways. So what PP2A's work is, when AKT is not in the environment, what PP2A does is it inhibits the activity of MEK and also ERK. So when their activity is inhibited, so it will decrease or minimize the formation of ERK. So what that gives us the idea is, when AKT and PP2A are not in the environment, tumor proliferation will happen, and when they are in the environment, they can control. So moving further, I'll hand over to Supriya.
Okay. Yeah. So in this plot of graph, what we are depicting is, since we know that SOS is one of the most important element in the pathway, because it is activated from EGFR and IGFR as together, as you can see from the pathway. So when there is when the dissociated SOS activates the EGFR, it helps to activate the downstream of the pathway, like the ROS, RAF, MEK, and ERK eventually. So we are understanding here the SOS functionality as well as the P90. From the graph, we can understand that P90 RSK is working as an inhibitor for SOS. So from the graph, we can see when there is an increase in ASOS, there is a minimal increase in P90, and after certain point we can see that the ASOS is decreasing. So that's the understanding we got from the graph, like from the pathway, that there are certain elements that helps to regulate it in a proper way, but in other aspect it helps to creation or activation of further elements in the downstream of the pathway. So this was the complete analysis we did of each element in the pathway. And going further, the only inquisitive questions we had was whether EGFR and IGFR together is working in ERK activation, or is it the EGFR only or the IGF-1R only that helps in the ERK activation. So in this plot, in this next section of code, what we are trying to understand is if we are given, if in the environment of cancer growth, if the only EGFR is present, does it involve ERK activation? If we have only IGFR, we do not have an EGFR involvement in it, will it activate the ERK pathway, ERK receptor? So from this thing, from this piece of graph, we can understand that with an increase in EGFR active in the environment, there is an increase in ERK activation, but then eventually it decreases. It doesn't demolish to zero, but it decreases eventually. But when it comes to IGF-1R, since there is no strong correlation between ERK, with time it does increases the value, but it doesn't produce ERK at all when we are considering only EGF-1R. Whereas when we are considering both, we can understand that both helps in the activation of ERK from this graph, that from the initial concentration it decreases, but still there is an presence of ERK in the environment. So this plot helps us understand that EGFR and IGF-1R together helps in the formation of ERK. It's not the only EGFR or not only the IGF-1R that helps in the activation of ERK. Moving on to the next piece of code, that is phase plane, nullines, and vector fields. Till now, we understood that EGFR and IGF-1R are the main receptors that are needed to in order to activate ERK. So to understand how they are interacting and what are the stability forms in order to create the ERK, we have to understand the vector fields related to it. So that's where we incorporated the phase plane and nullines functionality into our model. So this is the piece of code, and from all this thing, we understood these are the different scenarios and different hill functions which we considered to understand where is the stability, where we are getting the unstable, at what value of hill function and what value of threshold for each EGFR and IGFR we are getting the stability and the unstable. From this 4 plots, the first plot gives the stability because there is a co-expression of EGFR and IGFR. Here we can understand that at point, guys, can you see my cursor? Okay, so the 2 sections where it coincides, the red one and the blue lines, are the stable forms. The stable forms where each value are co-expressed. Similarly, in case of second also, we are just seeing that when EGFR, when we are increasing EGFR and we are decreasing IGFR, there is a kind of stability because they are codependent in some way, that it balances out the ERK production. But when we but in the third and fourth graph, we can see that if the values are extremities, like we have like high expression of EGFR, and when there is a low expression of IGF-1, there is a unstable environment. So the third and fourth depicts the unstable functionalities. The vector field is very unstable. So that is what we understood from our this piece of code. We tried to replicate most of the work from the paper which we are referring here. As you can see, these are the sample snapshots we have taken from the paper, and we tried to replicate most of it, but we couldn't do it. There are certain things that that need a lot of time. So we focused on most the important elements that were reproducible and that we could understand and make this as an educational app. So this is what the paper says, and we clearly did our best to make this replicate. As you can see, the null clients and the vector fields were also properly they were replicated to an extent. We are in a process of implementing sensitivity analysis because it's a complicated thing. We haven't gone through we initially thought of implementing it, but it's a complicated mathematical model that needs a lot of attention, and how to understand what is the sensitivity of EGFR on ERK and what is the sensitivity of IGF-1R on ERK. So it's a in progress thing. We haven't actually published it, but this is what we understood, that EGFR and IGFR co-expression leads to ERK. There are other pathways as well, which leads to ERK which leads to cancer cell proliferation, but majorly our app is based on ERK pathway. Yeah, that's all from our side. Please go ahead with the questions.
Okay, questions? What starts? You see the EGF, you see ERK come up and then go down again.
Mm-hmm.
What is turned on at the beginning of the simulation to turn on the ERK?
It is like from the graph, what we understood is there is an initial amount of there is a certain amount of EGFR and IGFR that is needed in order to activate the ERK, and this happens for a brief period of time. Supposedly, not supposedly, according to our analysis, we saw that it takes about 5 minutes to 30 minutes to completely activate the ERK, and then it decreases the value eventually. Like when the time frame increases from 30 minutes to like 40 minutes or like 60 minutes, there is no much activation in ERK happening because of EGFR and IGFR. It is the initial phase, the initial initial time, that is 5 to 30 minutes, that it takes to increase the ERK receptor. Sorry, the pathway.
But something has to be turned on at the beginning of the simulation. At the beginning, you have no ERK, right?
No, we have taken the wild type. So the initial concentration of EGFR we have taken is 8,000. So we are not starting from zero in this model.
No, but if I look at the time series, I see ERK 0, low, it comes up, and then it goes down again. What happens at the beginning of the simulation that starts the process?
From the model which we built, from the model which we used, we understood that there is an element of DSOS, the deactivated SOS, that binds to EGFR, which EGFR is not in activated state. This DSOS and EGFR when it binds to EGFR, it creates an activated EGFR. This activated EGFR gives results to activation of ASOS. So it's the plot says the EGFR active. It doesn't say the EGFR quantity or the concentration, the wild type, the active EGFR in the environment.
Well, but I'm a little confused about the terminology. You
Okay.
talk about things like EGFR,
Mhm.
but it's not EGFR that you're talking about.
Yeah, it is the EGFR active state in the environment.
Right.
So initially, EGFR active state will be 0 because the dissociated SOS is not bind to EGFR, the one which we are which is there in the environment. So once the DSOS is attached to EGFR, it gives rise to EGFR active. That EGFR, as we increase the EGFR active concentration, it binds to DSOS and give rise to EGFR active. That is why we are seeing the value of EGFR active from 0 to a certain level.
So I think it might be helpful just to try in your app to explain what's going on, because the arrow diagram, the pathway, the arrows you use aren't the ones we normally use. But secondly, it's not clear what happens at the beginning. In other words, if your SOS is turned on, so you start to imagine there isn't any SOS. Now you turn SOS on. That starts the process.
Yeah.
That would be clear, help things be clearer, perhaps. And then also explaining that when you say EGFR or IGF-1R, you don't mean the receptor, you mean the amount of activated receptor.
Yes, yes.
Because normally what you do in a simulation like this is you would change the amount of IGF or EGF in the environment and see what happens.
Okay.
But here you're changing the amount of receptor, and that's not something that one would normally be able to do in an experiment, right? In an experiment, you put the growth factor into the environment. You don't have the ability to control the amount of receptor directly. So I think that the model is very interesting, but you might want to try to be careful about the terminology. When you say words like expressed or regulated, those have specific biological meanings. And you, I think, want to be very careful to make sure that when you say terms like that, that they're in fact what's happening biologically, and that they're what's represented in the mathematics. I think the basic idea of what is activating what is quite, came across. But there were places that I was a bit confused about either the terminology or the process that you were simulating. And I think maybe you have a week now to revise things, so you maybe can go back and clarify a little bit of that. I don't know, maybe other people found that didn't find that confusing. I was a little bit confused by the analysis. The other thing I wanted to understand is you showed a null line, a phase portrait. Where did that come from?
This one you're talking about, Professor?
Yeah, how is that related to EGFR and IGFR?
So,
What am I seeing here?
so X in this is EGFR and Y is our IGF-1R. So the elaboration is done in the below part, where we have shown like what the red lines and the blue lines understand and what is X and Y doing in this graph.
Right, but how do you know that those are the relationships? In other words, where did the equations come from for your receptor activities?
This piece of information we got it from the paper as well. So
So these null lines, it's already there in the paper, and then we tried to replicate that.
So can you scroll up just a little bit? And I know we don't have too much time, so I don't want to take too much time. We can go over this more next week. But you write a pair of ODEs here for X and Y. X and Y represent EGFR, active EGFR, and active. I don't understand. How are they even regulated? The level of active EGFR or active IGFR is an input. What they're not downstream of anything. What controls them?
First, we are taking the value from the actual model which we have. Then we are passing those values to the differentiated values to get the null cline. Null clines are where we get the zero values. Dx by dt is 0. So first we are for each time frame we are getting a values for EGFR and IGFR, and then passing that values to our differentiated equation, which we are giving here.
Okay, but I guess I don't want to take too much time, but I'm trying to understand. You show first the level of active EGFR and IGF-1R.
Mm-hmm.
What are those downstream of? Those are inputs to the simulant model. There's nothing upstream. If you look at your pathway, there's nothing that points to them. Everything is downstream of them.
Yeah.
So they should have a constant value in your simulation. So I'm not sure what X and Y represent here in this model.
Okay.
How do X and Y relate to the big model? You explained the big model, but how did you reduce it to how did you come up with X and Y?
To be honest, we took it from the paper. So we tried to understand what the paper is saying and understand how EGFR and IGF-1 are interacting amongst each other. So so the values which we are passing to the differentiated this piece of equation is based on what we got from the paper.
But I guess, okay, so I'm a little nervous about having something in a notebook if you don't know what the derivation is, because if I look at EGFR and IGF-1R in your diagram, they're not symmetrical, right? They do different things.
Yeah.
Your pathways are not symmetrical. But here the two ODEs you have are symmetrical. I could replace X and Y with each other. It doesn't change anything.
It's not the concentration, but the signal transduction that happens in the pathway.
So maybe when you revise this, you could explain what this means.
Sure.
Because again, if I look at the pathway diagram, the pathway is highly asymmetrical. But here you have a completely symmetrical pair of ODEs, which are very nice, and your null lines are nice, but I don't see how they relate at all to the full model.
Okay.
We don't have time now, maybe, but maybe other people understood that, but I was confused by that.
We'll work on that for the next presentation. Thank you for your input.
Thank you.
Anybody else, comments, suggestions, ideas? Anyone? Okay, thank you. Then we'll move on.
Well, I have developed an educational app for Senescence, and in particular, the feedback loop associated with DNA damage response and ROS, or reactive oxygen species production. So one of the main focuses of my app is on teaching what each of those foci are and what are their relations with each other, and how does that feedback loop ultimately occur. So to give some biological background, senescence is an irreversible loss of replication and growth potential for a cell. And we'll talk mostly about some of the ways that it can occur in the introductory phase of the application, mainly focusing though within the notebook itself on ROS and ionizing radiation, which is just a means to experimentally or artificially induce that DNA damage. But even though damage to DNA can be potentially a way to cause a tumor, and the senescence is a way to stop that. Senescence may ultimately increase ROS, which means that it would cause age-related diseases, because age cells, or senescent cells, are more abundant in age patients. So the main hypothesis created by these researchers, which I'll discuss very shortly, is that very high DNA damage, or say having like 50% of the DNA compromised, is not really what causes senescence. Instead, it's just that the DNA can be continuously damaged by accumulation of ROS, and that is sufficient as long as that feedback loop occurs. So I've copied some of the learning objectives from my notebook, and I'll just read them off very shortly here. So by the end of completing an app, a user should be able to understand the positive feedback loop between DNA damage response that is necessary for cell senescence, and that was just discussed in the hypothesis. They should be able to describe the relationship between the proteins that control or lead the senescence, which include P21, P38, P53, and ATM. They should be able to suggest the consequences of senescence in the feedback loop, and also distinguish 3 possible cell fates. So the main proteins we're going to focus are ATM, which is a center kinase that detects the DNA damage. So that would be the very most upstream part of this feedback loop. Next is P21, which is actually quite down the line. And this is the one that is very much necessary for that feedback loop to occur. It is linked to P38 because it would activate it, and P38, when it's phosphorylated, or I shouldn't say that, rather. P21 has to activate GAD45, which is not mentioned here, and that would then phosphorylate P38, but we'll get to that. P38 is largely responsible for ROS production. And lastly, P38 has a very large role in determining cell fate because it is a transcription factor for so many different genes. So I copied a diagram from the paper and added a little bit of annotation here. So at the top, you have TP53 and CDKN1A. Those are just the genes for P53 and P21 respectively. And you see that on the left, you're having some DNA damage event that would cause P53 to activate. And once you go along the chain, you'll get some TGF beta near the end. So that, along with P38, will cause adverse effects on the mitochondria. We will not be focusing on those so much in this app, but the main consequence of that is that ROS production will increase from those mitochondria. That will then restart the cycle, causing more DNA damage on the left, and some of them being particularly responsible for growth arrest. So the primary resource I've pulled from is a paper titled Feedback between P21 and reactive oxygen production is necessary for cell senescence. I'll get into some of the limitations very shortly, but my main motivation behind choosing this particular paper is that it covers a lot of the proteins that are already discussed in the literature and have very important roles in signaling. But at the same time, the graphs that I have created are mostly a disjoint Z with theirs. So to give some model overview, I'll just highlight the step-by-step process with this pathway. So as said earlier, ionizing radiation is what we'll use to cause DNA damage. Then ATM will sense that DNA damage and interrupt the binding of p53 with MDM2, which is another protein that kind of helps to keep it under check. p53 will then activate the gene necessary to produce p21. Then that will bind to GAD45, activate the MAP kinase, which is p38, just an alias. Finally, that will increase ROS production, and we'll have a restart of that feedback loop, returning to step 1. So here I've included a very general or overview type graph where I just want to show that in my model, I will primarily be applying a dose of radiation of 20 grays at day 2. And you can see that one of the proteins, p53, spikes very quickly. And I mentioned earlier, it's one of the first to respond. It's very upstream. And in this case, things do level out to a steady state, but ROS, which is in red, is fairly high, which we'll discuss later on, that may be indicative of senescence. However, at the same time, those elevated ROS levels are not necessarily a guarantee of senescence, nor is there any particular biomarker within this model that says a cell is senescent. Nonetheless, it's primarily what I'll be relying on to indicate a senescent cell. And so, in addition, some of the other limitations to this model are that DNA damage and repair are highly dependent on only 2 very basic ODEs. One of them is just that DNA damage will repair it no matter what, and DNA damage is only dependent on ionizing radiation, or IR, and phosphorylated p38. So I'm going to leave this finding section very brief since I'll discuss it more in the notebook. But one of the main findings was that there's a bifurcation with ROS that I can use to be a biomarker for senescence. And additionally, there were sometimes some unexpected results, such as that ATM activation, decreasing the rate of ATM activation actually improves the overall result of senescence. But again, something I'll get into more detail in the notebook. So to move on to the notebook, I start off with a very basic introduction, which I kind of discussed before, about what senescence is and what some of the biological background is. So some of the hallmarks of it and the consequences are aging, as well as age-related diseases. Senescence is primarily a consequence of DNA damage, and that can occur exogenally, which would be outside of the cell, meaning some ionizing radiation that are applied with experiments, or just your everyday UV light. Meanwhile, endogenous DNA damage can be a factor because of uncapped telomeres, which means as a cell divides, its telomeres become shorter over time, as well as reactive oxygen species, which will be the primary focus of this app. So some of these running directives are included directly in the app, but I discussed them earlier. I also just discussed a very short guide on how to use the technology. In particular, I include with every plot a slider that would let you adjust the number of days in the simulation, but it doesn't affect the result, only what it looks like. So to get into the model itself, I first include 4 different plots, each of them showing the same plot I discussed earlier, but you can choose between it separately or together. So this just gives you a general idea of what the model looks like when there's no perturbation, and you might call it a control group, so to speak. So next up, I have an exercise designed to control the level of IR, or ionizing radiation, applied to the cell. So this is measured in grays, and if you apply only 5, so this is the default, ROS will increase very rapidly at the start of that event, but it ultimately goes to a steady state. I can drag it out a little more. This might be a small bug for me, editing before class. But conversely, if you were to increase the error intensity, then you'd have a higher steady state. And I discussed below that if, in case you did not see, then 8 grades of radiation is sufficient to push it into that higher steady state. So next, I've included the same kind of exercise, but instead it's just delay-based plot that will show different kicks to IR. And the purpose of this is just to show that a certain level of DNA damage is necessary for the feedback loop to begin, whereas in other cases, the foci will kind of die out and this cell's level of DNA damage should just become repaired. So I have one for P21, it's rather normal. P38 actually decreases in response to DNA damage. And lastly, P53 has a very sharp increase at the time of each event. So next I want to discuss why P38 was the one to decrease. And it's actually very simple. It's just because it is phosphorylated into P38P. So P38 has some roles in apoptosis and has its own signaling pathways. But generally speaking, it's upstream of transforming growth factor beta, and activation of either one of those will ultimately have an effect on the mitochondria that increases ROS production. So given that it's kind of an adverse effect, it would be best to try to inhibit it, right? So that's exactly what the next exercise is designed for. So in this case, I apply 40% inhibition, which would mean now P38's role in increasing ROS production is only 0.6 of what it used to be. And this is just the same as what the researchers proposed. So in the simulation, you can start out at day 6, which is 4 days after the radiation event. And if you were to see that, or try to customize some of those inhibition start times, then you might see different effects. I also include a separate exercise where I have only 0.15% inhibition, but I'll kind of speed through that for the sake of time. So next up, I'd like to return to P53, which was the protein that spiked very fast in response to the DNA damage. So the reason that happened is because it's directly, or has a very close relationship with ATM, which is a sensor kinase that is responsible for releasing the binds between P53 and MDM2, which helps to keep it in check in normal cases. So I've included a plot here where you can see that as soon as that radiation event occurs, the level of MDM2 will decrease very rapidly, or the bound one, I should say, meaning that those bonds are being separated. But shortly after, once everything returns to normal, it also returns to normal. So in other words, if the cell is not undergoing a lot of stress, then P3 it just becomes bound, and that'll help it to remain under control because it's in fact a transcription factor for roughly 500 different genes. P53 certainly has a lot of potential. So in the next exercise, I'd like to illustrate what happens if ATM is inhibited. So here I'm showing that if you were to increase the activation rate only slightly, you'd see very different results. Though I'm sorry this is rather out of the normal, but I think some edits just before class may have made a very big difference on my app. So I will rather go to the published version. It was only to fix typos, but maybe it had more impact.
Okay. Comments, questions? I'm sorry, did you show that you showed in your slide deck, you showed the feedback loop. Did you, basically the basic feedback loop that you analyzed, is that in the notebook too? You may have skipped past it quickly. Because it would be nice to be able to see the note, to see that diagram in the notebook.
This is
Yeah,
on
that's
the
that diagram.
abstract page.
Well, I might add that to the notebook too, so people can sort of track what's happening with the different components when you're going through the steps.
All right. So I'd like to return to this exercise here. So if the rate of ROS production by P38 is only 60% of what it used to be, then spacing out whether or not that, or what day the inhibition begins should have a large effect, right? But in fact, it doesn't. So
You mean the final outcome doesn't matter?
right,
You get
the final
same
steady state.
thing.
So this finding is consistent with the researchers who found that once treatment begins, it doesn't really matter so much about when it happens, but it can still have a very large effect compared to the control group on the right. So if you're 15 days in, then you're most likely in a deep senescent state, which means that it's already become irreversible, and you would expect that feedback loop to either continue or become interrupted by some inhibition like this. But yeah, I think it's worthwhile to go into this exercise, which I skipped initially, which is if you were to customize that inhibition rate, this is not exactly what was in the paper, but I wanted to see if there's a different result if you had only 15% inhibition. So in this case, spacing out the data start inhibition actually has a much greater effect. In this case, it's too late, 17 days in. But if you start it at day 4, which is only 2 days after the radiation event, or day 3, you see that it starts to go back down to that lower steady state, or ROS does, I should say.
So when you say it says 85%, but you mean 15% in terms of?
Yes, that is the bug I was fixing in the other version.
Oh, okay.
But again, I'm sorry that I
No,
broke too much in that.
no, I mean, it's okay. It's always a lot to clean up. The number of typos that are possible is large. I didn't mean to be, I mean, I suppose one thing you could do would be to have 2 sliders. One was the percentage inhibition, and one was the time at which you apply the inhibition.
Yes, but I think that could get complicated.
Might be hard to interpret. I understand.
So, since this is an educational app, I've included both hints before each exercise about what a user should expect coming into it, as well as sometimes I'll give explicit results such as these, or just general descriptions with the observations. So back to that exercise on P53 and ATMA. So if you were to increase the level of ATM activation rate, you'd expect that to be very good for this cell because it means more of those DNA damage response foci are activating and ultimately doing their job to repair the DNA. So let's try doing that. Before though, you should note that on the right, you have DNA damage. It's mostly low, but it's ultimately never going down. Its steady state is somewhere around 9. Meanwhile, if I were to increase the activation rate, it actually has a more adverse effect. You have slightly elevated levels over here. And if you were to do the opposite, which is to decrease that activation rate, you have an improved effect. The next plot is rather unnecessary. I have removed it in the other one. So I'd also like to illustrate that each of these DNA damage response foci that I have mentioned are very short-lived. So the reason that this feedback loop exists is because those foci are constantly being replenished, whereas they would normally degrade or become bound. So in this exercise, I've used some string manipulation to customize the number of events that are applied to the simulation. So here I have only one pulse as before of IR, but what happens if I were to increase that to 2? Then ROS and the DNA damage will shoot back up. Now it's most likely a senescent cell. However, I can also customize the number of days between those pulses. So that would give some cells some time to recover, let's say about a week. Now the cell is much better off, and that's indicative that some of those short-lived foci may have died off or returned back to their normal levels. So one weakness of that simulation, though, is that DNA damage could potentially be compounded. So even though it's a way to artificially induce that feedback loop, that is one weakness of it. So next, I'd like to illustrate the role of P21 and why it's kind of an essential link in this chain that I've been alluding to. So if we were to adjust the level of P21 degradation, then that should interrupt the simulation. So let's increase it, and indeed it does. So you saw before DNA damage was fairly high. Now it has gone about to 0. Now, at the end of my app, I've included some macroscopic effects. So those include its role on the mitochondria, decreasing its membrane potential, but also increasing the mass. And again, as mentioned earlier, some of those age-related diseases may be a consequence of senescence. But if you were to try to put forth some of these inhibitors that I've discussed in this app, it might be a double-edged sword because senescence is primarily a way to regulate tumors and prevent them from proliferating. So here I mentioned some of the limitations that I talked about in the slides, and I give a summary. So each of these proteins has very complicated rules. So I have only summarized them in very short lines about what their role is in this model. P21 has a very strong relationship with ROS, in fact, being one of the proteins that can induce its production. Next, P53 had a very strong role in deciding cell fate, but it was also the result of sensor kinases activating and being one of the first proteins to respond. Lastly, P21 is one of the essential links in the chain in this pathway. And I did not discuss the mathematical detail too much because it is an educational app, but one of the main points is just that everything in this app is very linear, meaning one thing leads to the next. Things are generally not dependent on multiple different proteins or factors. So to finish off, I've included some further reading. One of the points is that senescence may or may not be reversible, but even if you do manage to reverse it, quote unquote, then you might still have an inhibited or lesser growth potential and division potential. Also, I've left out a lot of the proteins and other genes that are essential for senescence to occur, such as GAD45. Those all have their own pathways as well, but you can only include so much in one educational app without it getting distracting. I want to finish off with a future work section again. So you can always have more proteins in your model, or you could take a completely different approach which is to only focus mainly on one protein for your model. Some of the proteins I left out are written here, one of them being a tumor inhibitor, and P16 has a particularly important role in that research I talked about earlier with trying to reverse senescence. Additionally, I did not account so much for basal ROS. It actually does increase DNA damage in this model, but it is something that I found could be excluded. Then lastly, I'd like to comment that DNA damage repair can be more sophisticated than the model proposed. For example, you could have some event where if the level of DNA damage gets too high, then perhaps the cell just dies. So that is all for my presentation, and I hope it was understandable.
Okay, questions, comments?
I have a question. I don't know the biology side of it, Pete, but is it possible for a cell to get multiple parts of its DNA to be damaged? Or if a part that is damaged doesn't affect how the cell would survive or die? And if that is something you think could be also done with this model by adding more complexity? It's great, but I'm just asking if those things are possible.
So I will admit that I don't know a lot of the background behind that myself, but the way I would probably think about it is to design several different parameters for the level of DNA damage. So if you think particular sections are most important, and you think that if those sections are compromised, then everything should shut down, then that could be one way to make the model more advanced.
Thank you.
Other questions? I was particularly intrigued by the demo section you did where you hit the cell with 2 low doses of ionizing radiation. And you showed that if you hit it with 1 dose, the cell recovered. But if you hit it with 2 doses too close together, it caused the cell to go into a senescent state. I thought that was really, really a great, the ability to change the number of pulses in the interval. Again, maybe changing the amplitude too, to ask the question. If the minimum amplitude to cause senescence with 1 pulse is such and such. If I have 2 pulses, then I can lower the amplitude if they're within a given distance. That I mean, it's more than you can do for this application, but you could do a little phase diagram where you have the interval between the pulses and the amplitude of the pulses, and then do you go to senescence or not? And that could be a really interesting plot. I think that there's somebody, a successor to you, could do a whole application just built around asking the question of what is the behavior.
Yeah, I had a lot of fun designing this plot.
I think that one really that's a very rich set of things to explore. If you have 3 pulses, what happens?
So that one would be indicative of senescence, but you might have to wait a really long time between each of those pulses in order for the cell to recover enough.
So that one, I think maybe if you could control the amplitude of the insult, that one having that second slider maybe as a separate plot, duplicate that and be able to make the amplitude of the IR a little bit less. And ask the question, here I start out with 3, I go to senescence. How much would I have to reduce the amplitude of the insult so that the cell recovered? That might be a really interesting thing to look at there.
I'm unsure about it. This is already a much smaller dosage. Ordinarily, I had 20 grays of radiation. This is only 5.
So 20 gray knocks you into senescence with one exposure, right? Or not?
No. Or it would, excuse me. 20 grays does, 5 does not.
Right. So yeah, so then 5 gray will knock you to senescence with 3 if they're close together. But maybe 2 gray, because you could ask the question, right? I go out into to sunbathe. How many days do I have to stay out of the sun to recover? And if I sunbathe once, twice, or 5 times, because here you're seeing that the effect of the damage is much more than cumulative. It's you get this all or nothing behavior, right? You either recover or you don't. It's really, really interesting. I think that's a fascinating plot. It's really a lot there to explore. Other comments, questions? I know we have to rush a little bit, but Peter, you're going to present again, right, in a week?
Yes, I suppose so.
So great, that's good. So we have time to dig in a little bit more. Okay. I think Joel and company are next.
Yes. Here, I'll share my screen. All right. Is everybody able to see everything okay?
Yes.
Cool. All right. In the interest of time, I'll kind of, because I know it's 6:15, so I'll kind of jump right in here. You've been seeing our work all semester, right? We've been talking, and you've also seen the unification of 2 groups. Joel has come and joined the dark side. He's come and allied with us. You know, first we were off talking about robotics and waving our hands around evolution, and now here we are talking about homeostatic cellular topologies. Isn't that a mouthful? But so let's get going here. So tissues are incredibly interesting, and in my opinion, they're just wonderful examples of collective behavior at its finest, right? So we get this, I mean, in my opinion, you know, cognitive science has got it wrong. We're skipping much ahead, going to a circuit of 10 billion neurons when we can't even understand how a paramethea behaves in its environment. So, but that's just me, you know, and if you're like me, Connor, Eden, and I think also Lindsay, you're a little bit on the radical side, but that's just how it goes. But so like what we've been seeing this whole semester, you know, we have metabolic networks, we have signaling networks, and we have gene regulatory networks. We can also make networks out of cell lineages. So if you were to imagine, instead of A, B, C, and D, if you were to imagine like stem cells, you know, some sort of transit amplifying or intermediate cell, and then the final cell being something like a differentiated cell right here. So some of the things on the end of our cell lineage here, you know, we can turn these into also signaling, or not signaling pathways, but more biological networks to understand them. And so these things can have, and we can also understand these ways. So many of the presentations here today have talked about growth factors, and these are regulatory factors that are excreted by cells, right? And so we can imagine each, you know, one of these having things in which they're inhibiting or exciting, but I'll get into more of that later. Other cell types, right? So we can imagine this being a stem cell, this being an intermediate cell, and it can either have positive or negative regulatory factors that inhibit or excite the growth of another cell population. And this is really important, right? So through both for our like just foundational understanding of biology, to also just our really, really important applications in regenerative medicine. I'm sure a lot of you guys are familiar with the work being done by Michael Levin and Sam Kriegman in that group. Super, super interesting stuff. A lot of really just fun stuff. And I think a lot of this is very foundational to that work. So here, we'll start to get a move on here. So we've been, as we've talked about with you guys throughout the semester, we've been replicating the work of Natalia Komarova. There's this paper that she wrote in 2013, and there's been a couple of extensions of the paper, but a lot of them build off of this very, very simplified model, right? So if we think about that previous let's see if I can go back. This previous thing, we can also just understand this in a more simplified sense. We can say that if this is, let's just imagine A and B here, right? This could be stem cells. This could be differentiated cells, right? These are just the two kind of differential equations that govern our simplified tissue. You know, in the most simple sense, our tissue can just consist of stem cells and differentiated cells, right? We know that it's going to have a rate of division, which we capture by L here. So these two forms of it are equivalent. So we can just look over here. We know it needs to capture a rate of division. We need to capture a probability that the stem cell differentiates, right? And we also need to capture the death rate of the terminally differentiated cells. So we know that stem cells basically have two major decisions. In this very simplified situation, they have two giant decisions. They can divide, right? And if they don't, then they just chill, right? They vibe out. If they do it at a rate L, which we capture in this differential equation up here. But it's also in the stem cells, right? Because we need to account for that. If they do divide, they do it at a rate L, and then they also get the opportunity to differentiate. And that's exemplified by this probability, right? So if they do differentiate, perfect. There's a new differentiated cell born, but they do this at the probability P, and then on the other side, we also, if they don't, then they have divided, so there's two of them, but that means they just chill, right? And we also need some sort of death rate in the system, right? So, and that's captured by, I also like this question, right? Die? Okay, here we go. So how does this actually end up working out in the model, right? So for the two-stage system, for the system that we just talked about, these two differential equations, there's really only two topologies, we call them, but they're basically the ways that we wire up the network that exhibit homeostasis, tissue homeostasis. So that, and those two situations are right here. It's when we have stem cells inhibiting their own division rate, and we have differentiated cells inhibiting stem cells' ability to differentiate. Or did I say stem cells inhibit their division rate? Differentiated cells inhibit the stem cells' differentiation rate, right? So you're saying, oh, well, what does that mean? You know, what does that mean, inhibit, or, you know, how does that actually work out with this equation, right? So we did a lot of work. You know, when Zach and I were slamming our heads about this for so much of the semester, we couldn't figure it out. Finally, we came crying to James, and we said, please help us out here. And we finally figured out the way in which, because we were operating under Lander's form, but we weren't finding results that we liked. But these are basically the three major groups of control functions that we found in the literature. There's variations, right? That's not like there's only these three, but these are the three that we kind of think are the most captured, right? The one that we're mostly going to use is this one by Komarova, but we also give a little bit of a demonstration about how it's used. So we can see, so let's first imagine a network with no control at all. Obviously it fails, right? There's nothing, it either fails or it explodes to infinity. Those are the only two options, right? So we need some sort of control because we know the tissues don't do this, right? So we need some sort of control. These are the two networks that give us that, right? And we also have activation here, just as an example, because I didn't point that out before. Now let's take a little bit more of a look at this. So we do a little bit more analysis on these two circuits. So we say, what happens when we perturb them, right? So what happens? So in this case, it's this same image that we saw. This is it, except at around 40 seconds or 40 time steps, we basically kill off all of the differentiated cells. And we can see this is very robust, right? And you know, and you might say like, oh my God, like when would that happen? And that does happen. Tissues like the olfactory epithelium, this is a prime example of something like that. You know, they're constantly getting perturbed. They're constantly getting the differentiated cells are dying off quite a lot. So we know that this is something that is something that happens in biology, right? So this model captures it quite nicely. But we also know that if we kill off all the stem cells, you know, if we were wanting this model to be biologically accurate, it should probably die because there would be no way for them to actually differentiate, right? These flow plots are simply our two state variables, stem cells and differentiated cells. It's just a single stable point. So what I want to point out to you really quickly is because since we're using Komarova's type control here, this parameter alpha traditionally she put K, but we put alpha here just so it was consistent, right? But this parameter kind of acts as wherever you, as you tweak this parameter back and forth, you basically either pull this population. So in this case, the alpha was set to 5, and we can see that the tissue's pretty much sitting exactly at that population, right? So as you turn K up, or as you turn that parameter up, so does the population go up. Just really quickly wanted to point that out. And we can see, so these are just represented by, in the flow, we can just see 2 stable points, really. That's all we see, right? The spiral character of this is simply just because this is in the complex world, right? So we're seeing this is just the imaginary number that's causing these beautiful spirals. But yeah, it's just a stable point. Very interesting. We can also look at the differences in the control. So all of these have a perturbation. This one, the one on the right is at around 50 time steps. The one on the left is at about 75. I think you can see why we kind of more went towards the Komarova control, or why we kind of preferred it. We had a little bit more. So in just toying around with the way that parameters reacted with these very simple systems, alpha and n also in the control, or the Hill control function, they react differently the more you tweak them. So in Komarova's version, they have a very simple relationship. The higher you tweak alpha, the higher the population rests. In Landry control, that is not the case with the parameter. The parameter acts in a very different way. Sometimes it's in Landry, as it gets more squished towards the origin. It doesn't drop to 0, but it gets more compressed. In Hill, it has an effect on the imaginary number of the eigenvalues as the trajectories become more rigid and they stop with their spiral type character. But they also end up, like in the Landry situation, since it's a very similar equation, they do get more compressed towards the origin. We can also see, but I mean, it's not like we're saying that Komarova is going to be the best no matter what, because there's certain situations in which it might not be, but in which we'll see some of those later. But I mean, we can see there's really interesting. I mean, look at the Hill one here. There's such a violent reaction by the amount of stem cells. And although we should definitely point out this is not a biologically accurate tissue, right? There wouldn't be more stem cells than differentiated cells. So that's something that we were kind of looking for. We see that that's at least kind of present here. You know, it's not to the type of correlation that we would want in this first model, but just thought that we would point it out, right? So these are our two circuits. A little bit of, so this isn't in Tellurium, but I was also exploring some of the dynamics of it, and I just figured I would show it to you guys. So, you know, you would think that these simple circuits would be, you know, we wouldn't see anything, right? We would just kind of see stable points out wherever you basically put the kind of carrying capacity at, they would reach that population and they would stay there, right? But it turns out, at least in this circuit, there's some interesting stuff. I mean, it got to the points where I found a Bogdanov-Takens point, which is a very high, basically a global type bifurcation that happens. So at first I found this Hopf bifurcation, which, you know, is characterized by a limit cycle around it, and I was trying to find it. But then I was just like, then I evaluated the eigenvalues at this point and basically realized, oh, okay, well, actually we have a 0-0 situation here, and this might be something different. Very interesting. You know, this is in negative probability space, so that's something to watch out for, but interesting nonetheless. This B is signified, so very, very briefly, in the interest of time, I'll just very quickly go over what this is. This is a branching bifurcation, this is a Hopf bifurcation, and this is a fold bifurcation. Connor and Eden talked a little bit about the fold bifurcations, and if you want me, I'm giving them a shout out now, you can go test that out and learn more in their notebook, because I think it's a great introduction to this stuff. But yeah, so what we see here on the Bogdanov-Takens point, by the way, is that as we tweak P, this dark line is all of the types of fold bifurcations we get. As we turn these parameters, we can see different types of bifurcations, fold bifurcations along this axis. As we then, as P sits at this value and we start to turn L, we can see these are all the, these are all the Hopf bifurcations. Very interesting stuff. All right. So I'll stop talking because I have a tendency to do that too much, and I'll switch it. I'll leave it up to who's next. Zach.
Sorry, just before you go away, so you have a stable limit. You have a stable limit cycle for,
So this is not so there's an unstable limit cycle, I'm sorry, in between these 2 points, right? Because we don't want to break existence and uniqueness, but there's an unstable limit cycle in between them. This is right around the point in which it happens. The limit cycle is just so fragile. It is like incredibly, it happens for like a split second. And it's very hard to get a hold of for me right now. I just figured that I would show this, but no, I'm sorry. I should have explained that.
Okay, well, thanks, Gabe, for that. I guess I'll take over. And so next we have the 3 compartments. So you can go ahead and switch to the next slide. So essentially, a lot of our work focuses on Komarova's work, particularly in her 2013 paper, where she did take these 2 compartment, you already saw the 2 compartment models from before, and then she decided to take it a step further and do a 3 compartment analysis. And from there, instead of just finding 2 stable control orientations, she found 21. And unfortunately, we're not going to go over all 21. I'm sorry. But what is interesting is that the first 2 in later work from Yang, Axelrod, and Komarova in 2017, they found that those first 2 result in biologically realistic tissue homeostasis. So we focused on those 2 that were actually, biologically relevant. And to focus and to mention one more thing, these all contained only 3 control loops, 1 from each cell type, and also they only used a constant death rate. So next slide, please. So in this slide, you can see us focusing on these 2 different control loop orientations, and basically, what you can see is that based off of having an event that we set at time 40, where we set I believe it was the stem cells to 0, everything was able to recover fairly well despite that. And you can see that, or actually, I'm sorry, it even says it in the title. I should be reading. Yeah, so one was eliminating the stem cells, one was eliminating the intermediate cells, and another one was eliminating the differentiated cells. And in all 3 approaches, they were still able to recover reasonably well, except for with the stem cells, obviously. Yes, thank you, Gabe. With the stem cells, obviously setting stem cells to a population of 0 will kill it off, because there is no way for it to continue the process of moving from stem cells all the way to differentiated cells. But then you can see below that, you can actually see the flows that we have. And so as opposed to in Gabe's flow, with the 2 compartment model, this one, since it's 3-dimensional, we use 3 different orientations, basically cross sections across the flow, to show you different aspects of the flow to get a better idea of that. And you can just basically see that there's a very clear attractor in this case, and that it falls right in there almost no matter what, unless you kill off all the stem cells. And then on the second one, on the second half, we have similar events going on. Again, it's able to stay consistent despite perturbation, and it goes right back to where you would expect it to be. I don't know why this one doesn't have, I'm not sure if someone can help me here. I'm not sure why there isn't the one with the stem cell being killed off.
So I can jump in.
Thank you.
If you remember this one, if you get it to 0, the number of stem cell to 0, you have that anomaly happening where everything basically doesn't work from the beginning. So it's not a clear, but this is enough to show how it's not biological meaningful. Just putting it close to 1, the number of differentiated cells or intermediate cells actually explode, which means that they're coming from nowhere, because they should be coming from stem cells.
Right, and I think that is being described more in our notebook as well. But also with that case, there are some more complexities with this. I don't really understand these graphs too well, but you can at least see that based off of perturbations, no matter what the perturbations are, they still reach that steady state again. And then again, you can also look at the flows as before, and we can see that in this case, as opposed where with the other ones, there's a very clear attractor in all directions. In this case, when you look in particular across the intermediate cells and the differentiated cells, which is the flow in the middle, you can see that they're actually being pushed off, or actually they're being pushed off into a huge number of intermediate cells that we don't have plotted here. So next slide. And now these are some interesting analyses from the actual 3 compartment models that I decided to take a look at. One in particular that's interesting is that when you move from 2 to 3 compartments, the level of dynamic variability increases significantly. In particular, just by adjusting our differentiation rates to 1/4 and 3/4. 1/4 being the stem to intermediate differentiation, and then 3/4 being the intermediate to differentiated. This is in contrast to the default, where it is normally at 1/2, 0.5, and that's what we set all of our parameters by default. We can see that if we look at a bifurcation scan across by adjusting the death rate to the population of intermediate cells, that depending on where you start out with, that can make a really significant difference to where you end up at. Now, mind you, this is not a I guess complete bifurcation scan in that they don't reach steady state. Oftentimes that causes errors, but these are population levels after going very far out. And so you can see that even just by changing the death rate very slightly, it causes a huge jump in where it will end up being, and where you start out can significantly affect where you end up. And you can see that with the flows on the bottom, that we also have a saddle node, a saddle point right here, where the intermediate cells continue to decrease until they reach a certain point where they've decreased enough that now the stem cells start increasing, and the intermediate cells slowly start increasing as well. So this is just interesting as well, that when you move when you add that third dimension to it, you can have really interesting dynamics without really doing that much work. Next slide. Okay, and then finally, another cool thing that was interesting is that when looking at that first network, if you actually look at the bifurcations in that space by looking along by adjusting K, in this case, K is actually the same thing as A, which was referred to as an alpha or an A value in the Lander paper, and in the Hill function, I believe it's also used as an A value. In the original Komarova paper, she uses a K value, but essentially these are the same sort of thing to a degree. Like they serve the same purpose, which is to adjust the magnitude of the control functions. And so as you adjust the control functions, you can actually see that there are two major major saddle points that occur. One is close to the 0 point, and another one occurs further out into space. And you can see that as you increase K, this saddle point increases along differentiated cells. So the number of differentiated cells where this saddle point occurs increases, and then also the number of intermediate cells is actually kind of bounded by approximately about 20 intermediate cells. But as you lower the K, eventually that point shoots out into infinity in the other direction. And that's actually shown off screen. You can't really see it here. The one other thing, and this is something that Gabe had to also do, is that you have to incorporate negative space, unfortunately. In this case, this would be negative population. You can just ignore that half, but as far as the analysis goes, the analysis won't work. Sometimes it just wouldn't work unless I allowed for negative space to occur. That's just a problem with the model, the way it is. But if you just ignore half of this little thing, just ignore the little those two saddle points on the right side, But interestingly enough, right next to it is an unstable point that actually is in positive space. And so these populations are pushed away from killing themselves off completely, which is, a good thing. So overall, very interesting dynamics. And we can also see two fold bifurcations here as well. One close to the beginning at near 0, 0, and another one that's way far out there in, I don't even know what that is, at a high K value. There's another fold bifurcation that occurs. So interesting stuff. Definitely interested in taking a look further at that. But yeah, it's very interesting how how descriptive these sorts of bifurcations are at bounding the populations in particular directions, and how they continue as you increase or decrease the populations. And I will leave this. I think we have another card.
Yes. Also, I'm just now noticing this almost looks like a higher dimensional bifurcation, the saddle node on a loop.
Yes. Oh,
The
yeah,
one
no,
where
it
there's
is.
two folds.
Yeah,
Yeah.
no,
Yeah.
and look at the one below it. The one below it. If you look at that one, look at that straight line. No, the diagonal. You see that
This one?
diagonal?
Yeah.
It continues almost perfectly. Like it picks up perfectly. And then if you look at those two on the top left one, those two saddle lines that like intersect near that fold bifurcation, they intersect perfectly on there. They actually cross like right through each other. So there's a lot of interesting stuff going on that I don't fully understand. I mean, we need to take like a higher level look at it to better understand it fully.
It seems like there's something almost global happening here.
Oh,
It's really interesting. It's cool.
Yeah. I haven't even taken a look at the limit cycles for this too. The limit cycles I think will also help unravel a lot of what's going on here. But yeah, now I will leave it off to Joel for our widgets.
All right, now that Gabe and Zach explained how we can analyze these models, right? Here comes the fun part of the user adjusting these parameters and finding out for themselves what these compartments can do, or these models can do. So the way we organize this is by giving the user the option to change the initial population. So they can be done on those sliding widgets over there, and then changing the division rate. So any initial parameter can be controlled by the user. And with every change, we also provide a flow dynamic analysis and a bifurcation analysis that can guide the user to make changes closer to that interesting points that Gabe and Zach was just showing. Next. Right. And also we want to give the option for the user to compare different mechanisms. My cat just tried to kill herself. And we can give a control based on Komorova as showed, or Lander or Hill. And every button of that I'll show on the live thing explains what is doing, what is the control behind it. So it's easy for the user to see, right? The next option we have here is also the changing of the value A. As you can see on the example over there, Komorova has a population divided by A, which would be a problem if that would be 0, right? You know that computers and anything else in the universe don't like division by 0, unless if you want to create a black hole in your living room. So we catch that with a try errors, and we let the people know that he's using it, that 0 doesn't combine with Komorova model. But Lander and Hill can accept 0, so that's why it's important for us to give the user that option if they want to explore that side. We then give them the control of what type of control parameter is doing on a specific places in the simulation. So for example, on the stem cell division, we can say it inhibits or promotes that specific relationship there. And we can also say which species is controlling it. Is it the stem cell controlling itself, or differentiated cell controlling its division? And that's done for the other two parameters too, right? The stem cell can differentiate as explained by Gabe, and we can also inhibit and promote those and tell which cell does that. On the next slide, we are going to see that the user can stipulate what is the max time for the simulation. That is important because sometimes we just see things happening as Zach showed or later times. So we want to give that option too for the user to see if the system stabilized over time in case of oscillatory behavior. And we also gave the option of creating an event. So the user just check the box of creating an event, put what time the event should occur, and what is the species of cell it wants to kill in case, which is the set default parameter to 0. Or who knows, maybe it's exploring something and put cells that grows out of nowhere without we knowing how the system responds to that is also possible. And the bifurcation diagram is control. We set a range of parameters. I just cropped because it was hard to do like a list as a show. I cannot click the snipping tool at the same time. So this is the 3 parameters that are involved there that people can choose to do the bifurcation analysis on, and the species. On the next slide, we see some results. So this is the result for the default. People are going to see that whenever they start the application, that is basically no control involved from any cell to any cell. So it's very boring. We want it to be boring so people can see cool stuff when they change some parameters. And it always there's always the potential of a no convergence. Since, for example, if we do stem cell promote its own division or the differentiation, and differentiated cell promotes the division, if there's 2 promotes, there's a high chance of it exploding to infinity. And we want to show that there is a convergence error there, and nonetheless produce some kind of information out of that. So we lower the time of simulation very small, showing how the system is behaving. Like in less than 1 second, we have 2,000 cells, 2,000 stem cells, which is making evident the type of behavior we're seeing here with its flow diagrams too. Now that we learn a little bit of the 2 compartments, we can go to the 3 compartments, which is basically the same structure, but we have more complexity. As you guys can see on this slide, we have an additional intermediary cell that the user also can change. And the rate of division is now both for stem cells and differentiated cells. So there is even the option of not having intermediary cells to divide. That can sometimes happen if depending on the differentiation from the stem cell on a specific type. So that is also captured on this model if we turn that to 0. And the differentiation probability, right? That is that value that tells, okay, stem cells has this chance to become a intermediary, or an intermediary has this amount of chance to become a fully differentiated cell. And the death rate, as Gabe showed, for Komorova, the death rate was constant throughout, but different results can be seen when we have that also being a variable. On the next slide, we're going to see some familiar things, but now a bigger amount of things that the user can change, right? Since we have more cells, we have more interactions happening between these cells, and we want to give the same control to the user. On the next slide, also the same thing, simulation time is controlled. We try to keep everything in the same structure so the user cannot have like any surprises when it's playing with the 3 or the 2 compartment model. And on the last one, Gabe? Yeah, On the tree, because of the amount of complexity, there are some errors that can happen that we didn't figure out yet, but all of them will throw a convergence error. So the first message in magenta over there will appear, but sometimes you can see like this code just telling you that is also happening a convergence error. Now that everything was explained, if I can share the live code and cross your fingers.
Just did it.
There you go. Okay. So the way the notebook is organized is basically the same way as this presentation was organized. We first give an introduction explaining why and what these cells are and how the paper of Komarova Lander tried to deal with population and the control parameters. We gave those the same examples, and here is the fun part, right, where we can change some parameters. I cheated a little bit and I changed on the backstage. And so you guys can see if I turn inhibit and inhibit, which is basically this one here. So I'm trying to replicate the same one over here. And we see that we have this type of response. If I create an event and give it a time, we have the response on the event. Okay, hold on a second, behave. Oh, okay. So it's back on this scrolling. We have to figure out a way of taking the scrolling away.
Just share it with the, do you want me to share mine? Because I have it with the scrolling off.
Sure, yes, please. I think if you go to code, right, that might fix
That
stuff.
also fixes it.
Yeah, okay. Here, I'll stop the share and you can share and I can comment. I don't want to take more time. It's very small. Do you think that's enough for the presentation, James? I can stop here in interest for Lindsay and Gran. Okay. So the same thing can be done, and if you play around with those values, Zach, you can show how things change. Yeah, we just wanted to show live for you guys that in fact it works.
That's trying to do a lot. Really, that's very ambitious to try to do that. It's interesting that the two circuits that she says in the end that she likes are both your trivial topology, which is S inhibits its own division, I inhibits its differentiation.
Right.
And then basically D isn't doing anything. D is controlling effectively the death rate of the eyes.
Right. Or even
yeah. So in a sense, the interesting topologies don't actually wind up being doing much. But she rolls the ball out.
The interesting thing though is that she applied that to a specific problem in biology, which is the column cells, not columnar cells. So it could be that other system shows different behaviors, right? That it could be that differentiated cells can send a signal directly to stem cells. But on her solution and on the problem that she was looking at, those were the ones that most like recapitulate. She even put it through an algorithm, and those were the results from the algorithm. That those two were better recapitulating it.
Okay. I mean, the only other thing I'd say is in the slides, you showed the face portraits, but you have to adjust the X and the Y ranges of the face portraits so you can see if you have your arrows going off to outside of the region of the face portrait, you don't know if there's a fixed point hiding somewhere here or if it's really blowing up. Your time series seemed to show that the values stayed bounded. So you have to be careful to adjust the field of view of the face portraits.
Right.
I ran into that when I was preparing the things for the class. There's some pathologies here with things like the null lines and the face portraits where it's easy to get confused because you push there's a fixed point, but it's outside of the field of view. You don't know whether you have a divergence or not. Like here, that middle one, just there on the screen, that there. right? There is in fact a fixed point up probably somewhere. Well, maybe not for this one. This one is unregulated. But it would be interesting to basically expand the differentiation plot to see whether it's controlled or not.
Yeah, or finding or defining a function that knows where the position of the points are, and then we adjust crop to that session, right? Yeah. The other thing that this model is doing, the widgets are doing, is that we are not limiting the control that she did, or the simplest kind of control. In this case, you can have stem cell controlling basically everything if you don't change those parameters on the species controlling over there on the right. So that's something that gives a little bit of more perspective of what people can do with this.
Now you might just let the user control the ranges in the phase portraits so that you can see where things are.
Yeah, the only problem with doing that is that already, for some reason, at least when pushing it as a NanoHub application, is that NanoHub seems to not like it as well as running this locally. And so if we expand, like the flows are the ones that take the longest amount of time for us to run. And by expanding it past 100 by 100, the amount of time just explodes. So that was the only thing with that. We added a simulation time to allow you to like adjust the amount of simulation, but even then there's only so much that it can do before it just kind of fries itself.
Anything else? I'm sorry we're late. Lindsay, what do you want to do? I mean, we're happy to stay and listen to you, but I don't know what your availability, you two of your availability is.
I mean, we're good. We're also presenting next week, so we can kind of do a little bit of an abridged version if that's what people would prefer.
No, I mean, you just because you were the last on the list doesn't mean you should have any less time than anyone else. As I said at the beginning, the problem with these is always people think they're not going to have enough to say, and then trying to keep it short is hard. So I appreciate everybody's patience. But before we jump on, were there any other questions or comments on the app we just heard about? No. I think that's it. Everybody's done very ambitious things this year, so it's. I appreciate all the effort that's going into it. Okay, so Lindsay, take it away then.
Sure, and like you just said, if anybody needs to go for any reason, go ahead. But so I'll share my screen here with our presentation. Can people see the large screen presentation? Did I do it right?
Yeah, we can see that.
Okay, cool. Oh, and it's all conclusion. That's not good. Can't spoil it. Okay. Okay, so our project is called Analyzing Correlation Structures in a Model of Neural Activity Dependent Homeostatic Plasticity, which again is a mouthful. It's not currently up on NanoHub because NanoHub does not like the combination of the widgets, which I designed in Google Colaboratory, and the 3D plotting software that I am using. We think that's the problem. We're pretty sure it's on like NanoHub's end, but it's a pretty deep problem that Grant and Giuliano have been doing the bulk of the work to try and figure out. So our ambition is still to put it on NanoHub, but it's not there yet. It is publicly available, excuse me, on Google Colab at that link that I just sent in the chat. So if you want to mess with it yourself, you can go there and then just put a copy of it into your own Google Drive and work with it that way. So first, a little bit of the biological background. I this is all sort of basic like neuroscience stuff. But I'm not sure if anybody's familiar with neuroscience, so I'll go over the things that are most important to know. So basically, ions are normally not allowed to flow across the membrane of a cell, any cell, because they're too charged. But there are special proteins that are embedded into membranes called ion channels, which do allow particular species of ions to flow across. And when those channels are present, ions will flow according to two things. One of them is their concentration gradient. So if there are more ions of that type on the outside of the cell, they will tend to want to flow inside. And then the other thing is their charge gradient. So if the inside of the cell is more hyperpolarized, positive ions will want to flow in. But the other thing is that those two things interact. So sometimes, even though these positive ions want to be on the negative side of the cell, there are just like way too many positive ions already there. So the net driving force in this case is actually outwards for these ions. And basically all that you need to know in reference to this is that due to this balance between the concentration gradient and the charge gradient, some ion channels, their presence tends to depolarize the cell when they allow the ions to flow through them. And then other species of ion channels tend to hyperpolarize the cell. So that's just the basics. I haven't said anything about this activity-dependent homeostatic plasticity yet. So what is activity-dependent homeostatic plasticity? So we know that cells which are too active or too inactive for long periods of time will tend to adjust the number of ion channels embedded in their membrane in order to regularize their activity level. So as an example, and this is pulled from the paper that we are basing our analysis off of. In this example, we have a neuron. And this neuron is depolarized. It's like too active, and it's been a while that this has been going on, which has caused calcium to enter the cell in large amounts, which is what happens when a cell is depolarized. This causes, in the process known as activity-dependent homeostatic plasticity, this causes differences in the gene expression for different ion channels that the cell has. So in this case, it's too depolarized, so it's going to inhibit the production of those further depolarizing ion channels, and it's going to increase the production of those hyperpolarizing ion channels to try and even out its activity over time. So with that general framework in mind, I'm just going to briefly go over the specific model that we implemented, which was the first model in the paper that we were trying to replicate. It's the most simple one that they did. So there are 5 variables model, of the VM, which is the membrane voltage, the concentration of calcium, which basically tracks the membrane voltage. It's an indicator of the membrane voltage, and 3 species of ion channel, G1, G2, and G3. And there's differential equations for all of these, the form of which is just very standard in modeling these types of ionic, electrically excitable cells. And then some key parameters, which we will be playing with throughout the rest of the model. Each of the 3 ion channel species has a tau associated with it, a time constant, nominally. Basically, each tau tells the cell how should the level of G1 respond to your activity level, your calcium concentration. Should you upregulate G1 when the cell is too hyperpolarized? Should you upregulate it when the cell is too depolarized? And how much? And these taus exist for every one of these 3 ion channels a set of 3 taus. A couple of assumptions that the model makes, which are obviously not biologically realistic because this model is not really trying to be that biologically realistic. We're assuming that there's only 3 species, that none of them are voltage gated, so you're not going to get any action potentials or anything in this quote unquote neuron. And we're greatly abstracting the process by which calcium concentration gets translated into ion channel expression and incorporation into the membrane. Which brings me to the procedure that was done by the paper. This paper is by O'Leary et al. 2013. So basically they generated this model and they generated a bunch of initial conditions. So a bunch of cells, each of which had some existing number of G1 channels, G2 channels, and G3, placing it on a point in 3D G1, G2, G3 space. So this cluster, I don't know if you can see my cursor, but I'm looking at figure D. This cluster of orange points is a cluster of initial conditions. And then to this cluster of points, they applied 3 different versions of activity-dependent homeostatic plasticity. The first version, all of the taus made sense. So all of the ion channels which were depolarizing naturally would be downregulated when the cell was too depolarized, and conversely for the hyperpolarization. That's the blue points. And then in the green situation, they just changed the magnitude at which each of those processes occurred. And then, but in the red situation, they completely flipped the sign of one of the taus, meaning that one of the regulation rules, which used to make sense, now doesn't make sense anymore. Like you wouldn't expect it to work, but they showed that it does. What happens to each of these initial points is that the values of G1, G2, and G3 are changed such that the cell's target activity level is again achieved. And then they observed that depending on which of the homeostatic plasticity rules they applied, a different cluster of these final points emerged, and they seemed very separated, and they seemed to have different pairwise correlation structures within each group. And so they concluded that the correlation structures emerge from the different homeostatic plasticity rules, and that the specifics of the correlations depends on the specifics of the rules, yada, yada, yada. This is a quote from what they said exactly. However, we suspected that this might have been not because of any fundamental reason that the rules cause the correlation structure or anything like that, but simply an artifact of the fact that they chose the initial conditions from a very small area of parameter space, and that the different sets of rules were defining a direction of movement in this parameter space. But there's no reason that that should result in correlation structure persisting over time. So one of the main goals of our application was to demonstrate that their conclusion was in fact an artifact of the initial conditions that they chose. So with that sort of broad, really broad and probably too fast motivation, I will hand it over to Grant to explain the outline of our notebook.
Okay, so I kind of set this up kind of weird. So first I'm going to go over the entire outline of our notebook in these slides, and then we're going to transition over to actually looking at the notebook where hopefully you guys can follow along. It is linked in the chat. So the way we set up our notebook was first we just established what the learning objectives of the entire notebook is, and then we repeat that background that Lindsay just gave. so that people kind of know what they're getting into with this and what it is that we hope to build upon with the paper that we were working from. And then we give a description of how this model is set up, all the different differential equations, and why they are explained to make sense in the context of the paper. And then we have 3 different modules. So the first one is to give the user of the notebook kind of an intuition of to how this model works. And it just works with one neuron and shows how the parameter space changes over time. And then after that, we do we let the user define their own parameters to simulate many neurons at a time. And they can observe the correlation structure that was purported in the paper. And they also have the opportunity to change these variables and see how, if they can, break that essentially. And then module C is simply allows the user to analyze, usually using pairized correlation graphs, the pairized correlation of what they just plotted. So it's based on what the user puts in. And then at the end, they are prompted to try again with different initial conditions that we find to break the correlation structure that was reported in the original paper. So you can go to the next slide, Lindsay. So here's a little slide preview of that initial module A. We allowed the user to plot all 3 of the types of models that the paper had, which was the original taus, the flip taus, and the scale taus. And then they're also able to plot their own custom taus for one neuron if they want to do that. And so there's also some explanations given there. They're kind of just tiny here. You'll see them in the notebook. That gives the user kind of an intuition as to why this makes sense. Next slide. And then here I just wrote, I just wanted to show the how the model is defined. So we have in our notebook here all the differential equations that are used for the state parameters, and some explanations behind the biological basis of them, like Lindsay explained previously. And so the entire point of this is to make the user aware of why the model, why the original paper believed the model to be a good explanation at first, because we do think that it works in this small parameter space, right? But we want to show that it doesn't work in a larger parameter space. Next slide. And so when we were making this notebook, we had a lot of problems with Tellurium and the plotting software, or the plotting library, as Lindsay said. So for our model, we actually didn't use Tellurium. We just defined a model in Python and NumPy. But we were able to get the widgets working, as well as the Plotly graph, which makes the graphs interactable, which is pretty important because, as you're going to see, in order to observe this correlation structure in 3D, it helps a lot to be able to move the graph around dynamically. And then module B also, this is where they're able to plot many simulation of many neurons at once with whatever initial parameter values that they want. And one of them that's of particular importance is the variance, which starts out as initially low. And when that setting is low, you'll see that the correlation structure that was found in the paper does seem to work for many different initial parameters. But as you increase the initial variance, you'll find that that correlation structure begins to break down. And then we also provided a graph that plots the trajectories of the parameter space, which has some interesting results, especially for the scale taus, I believe. And then here's the preview of module C, where we have these special graphs so that they can actually quantitatively measure the correlation of those final points.
Yeah, one thing I wanted to mention here is that I messed around a lot with how best to show this. The paper doesn't actually do any quantitative analysis on what the correlation structure is and how it actually differs between the three cases. They just kind of visually observe, like, yeah, they're different, therefore. So they do plot these pairwise correlation graphs. But I wanted to add a little matrix down here showing the strengths, because you might expect that even if the correlation directions are the same between the small initial condition case and the high variance case, they might not be as strong in one case or the other. I'm not sure exactly how successful that's been. It's certainly not a striking result as much as the visually looking at the plots is. So in future, I might mess around with some other, excuse me, I'm sorry, some other measures of how to quantitatively define this, like clustering algorithms, information theory, machine learning, something like that. But anyway.
So let's transition over to the notebook here. If Lindsay, if you stop sharing your screen, I could go ahead and share the notebook that I brought up. Okay, can everyone see the notebook? Okay, so I did already just give kind of the brief outline of what's in here through the slides, but so hopefully you guys are able to bring this up on your machines. And so we do provide some graphics inside the notebook to help the user with what Lindsay was able to explain earlier. And then we define the model in terms of the differential equations. And then if you're running this, the one kind of weird quirk of Tellurium, which again makes me want to just completely eliminate Tellurium from the project. And we did actually define a non-Tellurium version of this module, if that doesn't work for you. But in order to get this to work for you, you need to run the first code block and then restart the kernel and then continue on. And then it should work for you if you want to run the Tellurium version of module A. Otherwise, you can run the second version. So here's module A for the Tellurium version. And so it's interactive, and they can also plot their custom taus if they want to try new things. And then we have a non-Tellurium version. And so this is the graphic from the paper that shows the correlation structure of the final points. And this is right before we let the user do it for themselves, so that they can see kind of what is expected to be seen, and then so they can compare that to what they actually see. And then here I've already run a simulation. These simulations, depending on how many initial points you have, can take a while. So that's why I pre-ran it. And then here I did a low variance so that you'll actually will see the correlation structure. And then after you click simulate here, you're able to plot. And this is our plotly plot, which is great because it's a little bit confusing if you're looking at it from the wrong angle, but you can see that correlation structure if you bring it to the right angle. Where here the orange points are the initial points, just as in the paper, and these are the final points. And here's the trajectory plot, which you can also rotate.
Sorry, blue and green are different. You've changed the sign of one of your taus between blue and green.
In this case, it's a custom set that Grant has
Yeah,
defined on
there
the top.
isn't there isn't any particular biological significance to this choice here.
But what's the difference between the blue and the green trajectories?
Can you go back up to where you defined the numbers again for the custom? Okay, so the blue has values of 6,000, negative 4,000, negative 1,000, I think. So these have all the same signs, but just different magnitudes between the blue and the black.
So they're different tau values between blue and black.
Yes, yeah.
And the tau, as you say, so tau is the rate at which the adaptation happens. So a negative, it's the speed of adaptation. So a negative tau means you're moving away from the adapted state.
A negative tau would mean that an increase in calcium concentration decreases that channel. And a positive tau would mean the opposite, I think. I'd have to double check with the way that the equations are set up.
So depending on whether that channel increases or decreases calcium, the positive or the negative tau could be the thing that stabilizes, that basically restores homeostasis.
Right, yeah. Even in the case where, like I said, it doesn't make sense. It's the flipped case. It's coded as the flipped case throughout this notebook. That still brings you back to your target calcium concentration.
So if the channel acts to increase calcium levels, then you want to have the amount of that channel decrease to keep the calcium level the same. And so you need the tau to have the right sign to do, okay, I get it.
You would think, but it turns out that even if that's not the case, it still works.
That's which is very odd. I mean, this is in a sense, in the end, it's very similar to the Komarova paper, isn't it? I mean, you're basically doing homeostasis with control in a very similar way to the one that the Komarova team.
Do I hear another group merger is in the works?
You guys have pretty bifurcation plots, so sure, come on board.
We'll accept everybody.
And then we have module C with
there are pieces of the model that are different, but at some level, there's more similarity between those two projects than I would have guessed when you started out.
Okay, and then we just have that these pairwise correlation graphs as was shown before for whatever the user simulated, so they can see their own data. And then after this, pretty importantly, we want the user to go back and change these initial conditions, because the way it's set up, the first time you run it, if you don't change anything, you will see the behavior that was explained in the paper of the very strong correlation structure of the final points. However, when you go back and you increase the variance, if you can get this, then you start to see a breaking of this correlation structure. For this particular one that I just randomly chose, a lot of it is preserved, but then we also have it breaking here. And so that's kind of the most important point of all of this. And then after this, we just state our conclusion here, which do you want to do that here or in the slides, Lindsay?
Probably back in the slides.
Okay.
I think it's more concise over there. Which you can, okay, I will share it again then.
Oh yeah, I could have done that, sorry.
Okay, this time I do want to start with the conclusion. All right, so as you've seen, we were able to replicate the model from O'Leary et al.'s paper. There are differences in exactly where our clusters ended up and exactly where their clusters ended up, which are strange, but I'm pretty sure we're the ones that are correct. So I plan to contact O'Leary about that potentially and see if we can get some clarification and maybe share some code. But we replicated their main finding, which was that correlation structures emerge depending on the homeostatic rules that you set. And we also showed that it breaks down when the initial condition variance is increased. So, all in all, we find that correlation structure may still exist, but if it does, that is due to the inherent degeneracy of the parameter space rather than the ADHP rules themselves. So some future questions that we propose at the end of the notebook. One of them is what I mentioned earlier about some other potential measures to numerically demonstrate the difference in correlation structure. One of the other things we could do is map the basin of attraction of, you saw that all the points sort of aggregated on that plane. So that plane is a plane of steady state points for this model. So we could map the basin of attraction of that plane and see how that differs between different sets of homeostatic rules. We could investigate the role of noise in the system. And then we could also try and find points which I suspect exist, but I haven't seen many of, in which the calcium concentration blows up to infinity, or the number of ion channels regresses to 0, which could be interesting things to avoid. But yeah, I think that's all we had.
Comments, questions? I already asked a question or two. I think everyone did a good job and worked a lot. I mean, this is clearly an enormous amount of effort for everybody, and I'm very impressed. One thing I would say when you're doing revisions for projects, that depending on who and where people were in the project, I think there are a couple of people were tried to lay out very clear learning objectives, and I think that's helpful if you can. Not everybody will be able to do it, but if you could define the clear set of learning objectives and to exercise results in your notebooks, it probably helps make things more accessible. Again, depending on the project, some of these projects may demand a higher level of sophistication of the user than others. And so it's up to you to decide what level of sophistication your target audience is. That's your choice. But that might be something to help as you refine things to clear up a little bit for final revisions. I want to thank everybody. That was a long day. Everybody did a great job.